National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Good morning, ladies and gentlemen. Thank you to those who have joined the Zoom room and also official YouTube BRIN Indonesia for the second day of ISIBio 2022. I'm Aulia, will be your master of ceremony for ISIBio. Ladies and gentlemen, before we begin the conference today, allow me to read the guidelines of the conference. Presenters and participants are expected to join at least 30 minutes before the conference started. All participants, both presenters and non-presenters, are expected to join as much as possible all sessions in this conference. Please use the virtual background provided by the official committee. Also, please rename your Zoom username according to the following formats. For presenters, you can write down code of abstract or paper _name_institutions. For the example, P4.3 Octaviana _BRIN. Or for non-presenters, you can write down P_name_institutions. As the example, Octaviana _BRIN. Please mute the microphone, especially during the presentations of the keynote speakers. And in the parallel sessions, presenters should activate the camera during their presentations. Questions from all participants are delivered using the raise hand, Q&A, or chat feature in Zoom meeting. And when Q&A or chat feature selected, please deliver your questions according to this following format: name, institutions, the questions or the name of speakers, and also your questions. Every session will be led by a moderator, and moderator will decide which questions is to be discussed or answered. All participants, presenters, and non-presenters should fill the list of attendees via the link given in each session. Attendees are allowed to know or are not allowed to write down opinion or informations that is offensive, racist, or immortal in nature. An organizing committee have the full right to block or remove any attendee who breaks the above rules. Ladies and gentlemen, we will begin the event. Honorable Dr. Iman Hidayat, Chairman of Research Organizations for Life Sciences and Environment, National Research and Innovation Agency, the Republic of Indonesia. Honorable Head of Research Center for Applied Microbiology BRIN, Head of Research Center for Genetic Engineering BRIN, Head of Research Center under Research Organizations for Life Sciences and Environment BRIN, distinguished speakers and moderators, representatives from ministries, institutions, and universities, and all distinguished guests, ladies and gentlemen. Very good morning from Jakarta. Such a great time for me to welcome back every one of you to the second day of the 9th International Symposium of Innovative Bioproductions Indonesia on Biotechnology and Bioengineering 2022 ISIBio. Ladies and gentlemen, ISIBio has been held annually since 2012, and this year is proudly organized by Research Organizations for Life Sciences and Environment BRIN, bringing the theme of strengthening for bioeconomy through applied biotechnology, bioengineering, and biodiversity. This conference is collaborations with Indonesian Biotechnology Consortium and also ASEAN Federations of Biotechnology Indonesia, and supported by PT Berkaniaga Medica and Nutrilab. As the initial of the agenda for today is keynote speech, therefore may I invite Dr. Puspita Lisdianti as our moderator to lead this morning's sessions. But before that, allow me to read Dr. Puspita Lisdianti's profile. Dr. Puspita Lisdianti is a senior researcher at Biosystematic and Evolution BRIN that has interests on biofilm for health and environment, marine biomicrobiology, cacao fermentations, and also functional foods. She obtained her bachelor degree of molecular biology and agricultural chemistry at Tokyo University of Agriculture, Japan, and continued her master and also PhD at the same university, both majoring biosystematics and agricultural chemistry. Throughout her career, she is also awarded Habibie Prize at 2020, Satya Lencana Karya Satya 30 in 2013, and also Satya Lencana Pembangunan in microbiology. Ladies and gentlemen, please welcome Dr. Puspita Lisdianti. Ibu, time is yours to lead the plenary sessions.
Thank you very much, MC Mbak Aulia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Honorable Chairman of Research Organization of Life Sciences and Environments, Director of Research Center for Applied Microbiology, Genetic Engineering, Biosystematics and Evaluation BRINs, Chairman of this symposium, and all of the committee. Also, honorable speakers and all participants in this symposium, ladies and gentlemen, I am Puspita Lisdianti, the moderator of this morning symposium. Our keynote speaker today, the first is Dr. Muhammad Sahlan. He is as a representative of Professor Heri Hermansyah, the President of ASEAN Federation of Biotechnology Indonesian Chapter and Dean of Faculty of Teknik Indonesia University. Let me to read Dr. Sahlan CV briefly. Dr. Muhammad Sahlan is a researcher at the Bioprocess Technology Department of Chemical Engineering Universitas Indonesia, obtained a doctor and master degree at the University of Agriculture and Technology, Japan in the field of protein engineering, obtained a bachelor degree at the ITB in the field of chemistry, awarded first winner of invention in beekeeping field, ASEAN Apiculture in 2018, top 107 Indonesian Innovator Business Innovation Center in 2017, gold medal IGEM, In 2014, Roseno Award, Roseno Octroi Bureau, and the National Spice Board in 2013, and Superior Researcher Indo Food Research Nugraha in 2011. Research interest is waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and PICO remediation. His Scopus H index is 14. Before the presentation starts, let me inform you how the presentation will go. First, Dr. Sahlan will be invited to present the material for 30 minutes. After that, there will be a question and answer session for 10 minutes, followed by conclusion. Now, allow me to welcome Dr. Sahlan with the topic Development of Research Collaboration between BRIN and Universities on the topic of biotechnology, bioprocess to improve the bioeconomy sector. Dr. Sahlan, time is yours.
Thank you very much, Dr. Puspita, and also the Master of Ceremony, Ms. Audia. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. I think I have to share my screen for the presentations. Let me to share. Did you see our screen?
Yes, sir. It's already full screen. Thank you very much.
Thank you very much. Okay, recently, now I'm a representative of Professor Harry Hermansyah as the Dean because he have another schedule in this time, and he implied maybe today in this time. So I'm sorry he could not present his presentation. Okay, as Dr. Puspita introduced that in this time, I would like to present about the development of research collaborations and between BRIN and universities on the topic of biotechnology, bioprocess to improve the bioeconomy. In this time, I will focus the presentation or the universities in about the Faculty of Engineering, Universitas Indonesia in collaboration with the BRIN. Recently, I'm also head of the Entrepreneur Innovations Unit and also Reverse Engineering Center that have the correlation between this presentation. Okay, talk about our Faculty of Engineering. We already established on 17 July 1964, and in that time we established 3 departments: civil engineering, mechanical engineering, and electrical engineering. Continue 1 year later, we also established 2 departments: metallurgy and material engineering and architecture. In 1985, we also established the chemical engineering department, started by petrochemical department in that time. And in the 1999, we also established the industrial engineering. Recently, we have a 13 undergraduate program, 2 professional program, 12 master program, and 7 doctoral program. So in detail about the study program in Faculty of Engineering Universitas Indonesia, we have undergraduate program about a certain regular program and 11 parallel program and 7 international program. In the professional program, we have 2 professional program. This is for engineer and also architect. And for the master program, we have 12 master program and 7 doctoral program. So this is the detail of the 13 undergraduate study program from the civil engineering and also mechanical engineering. This is the first in study program in Faculty of Engineering. So all the 7 departments, we have the undergraduate and also international undergraduate program. The international undergraduate program, we have the single and double degree. And every department, we have at least another 1 study program. For example, for the chemical engineering, we have also bioprocess engineering study program. That this study program have very related with this topic about the collaboration between BRIN and university in the field of the bioprocess and biotechnology. And also related to the bio, we also have the biomedical engineering. This is for the postgraduate or postgraduate study program. We have 21. For the professional program, we have engineer and architect. For the master program, we have the civil, mechanical, electrical, metallurgy, and also environmental engineering. We also have the master program for this field. And for doctoral program, we have a 7 doctoral program. This is related to the department in Faculty of Engineering. So since this year, our dean, Professor Halihan Mansa, he's the new dean of the Faculty of Engineering, and he know that the condition in the world is very accelerate and the change, and also we have make the strategy how to challenge the accelerated change. So this is the condition in our world. The first is about the volatility of the world, uncertainty, complexity, and also ambiguity of the condition in the world. So based on this condition, we need to collaborate and partnership with other institutions, with other stakeholders. We know that we have the now in disruptive era and also new normal. We have global competition and national competitiveness. We have also industry 4.0 and digitalization, and we need the multidiscipline how to challenge our world in recent condition. So we initiate this challenge by the name of the Entrepreneur Initiative. We have to challenge. We have the good input in our Faculty of Engineering. We have the students, we have facilities, and others input in our Faculty of Engineering. So we have to make the good impact. We have to increase our service excellence, customer satisfaction culture, reputations, glory on the nations, and also revenue for the economic and advancement. So based on these conditions, we decide 3 strategies. The first is empowering engineering entrepreneurship. The second is impactful research and innovations. The third is modernization of engineering education. Based on the 3 strategies, we have 11 excellent priority programs. The first is the organization of multidiscipline engineering, reverse engineering center, engineering seed fund and grant, engineering revenue recognitions, database engineering networks, organization strategic engineering educations. So because we focus on also in educations, we have to increase the quality of the organizations, and also virtual engineering education facility. As you know that the new era need this conditions. And engineering professional program partnership and collaboration package, international academic recognition and engineering student entrepreneur program. So by this 11 excellent priority program, in this time, we focus on the impactful research and innovations by using reverse engineering strategy. So for the impactful research and innovations, we have a platform for the multidiscipline technology development and product co-creation and reverse engineering. This is the new center in Faculty of Engineering Universitas Indonesia. We have to collaborate with the alumni, society, community, and media, government, business, and also industry. So we have the researcher, we have the facilities and also students, we have inventions, fundamental research funding, and also for the local and global market, we have to innovative solutions, and again, revenue income to increase our quality of the output, and asset equity and also license of the intellectual properties. Based on this strategy, we have also the product after we use this strategy in reverse engineering. We learned from this pandemic, COVID-19. We have one good example, in this case, about the ventilator. So by reverse engineering, we have an activity to increase the value of the technology product, either by modifying or by adding features of the technology. For example, in the first 2000s, in the first time, we make the laboratory prototyping one in this stage, and then after about 6 months, we succeed to use to make the prototype 2, and after 1 year, we succeed to production in industry. So this is one of the good example from the laboratorium or from the lab to the industry. We study the original product, and we analysis the product and engineer. We make the prototype and we establish the new product by this scheme of the reverse engineering. By this reverse engineering, we make this center. In this center, we collect the demand from the industry and also from the community. We design the product, and we make the seed fund also. We give the fund for the researcher and also for the team, from the researcher and also from the students that want to make the reverse reverse engineering product, and the output is the intellectual properties. Yeah, we synergy with our another institution is Lemtech, with the alumni and partnership industry and sponsor. And with another scheme and the license, we also make the startup and incubate for the technology in implementations. And we hope we can increase the revenue of the market revenue for the Faculty of Engineering. This is another product that we have succeed in the pandemic era. Especially, this is the product for in health, because in these conditions, the product of health is very useful or very good in the market. The first is the COPN20. COPN20 ventilator is the first succeed product, and this product is collaborate not only Faculty of Engineering, but another institution and also the government, how to develop the product and accelerate this product from the laboratory to the industry or to the market. The another product is air purifier with non-thermal called plasma method. This is also the reverse engineering product from Professor Bismo that he expert in the plasma technology and apply this plasma to the air purifier. And this product is also already licensed to the company in Indonesia. Another product is gas supportable incubator. Is a little bit different with another product. These implementations in the society is the scheme is by the community engagement. This product is the incubator to warm premature babies or low weight birth babies in order to prevent hypothermia. And this product is portable design and very simple for mobilization. That very good with the Indonesian situation. And this is another product for the implant, and another product is like propolis, and in traditional traditional medicine. This is also from the chemical engineering. So I would like to talk about the example of collaboration between Faculty of Engineering and BRIN. This case is about the development of dengue paper diagnostic. As you know that the dengue is common in tropical countries, including in Indonesia. As we know that about 390 million of the people is already infected by the dengue, and about 75% of the citizens in the tropical countries have infected by this disease. So in the 2019, it's about 120,000 people infected. It is decreased in 2020, but I think it's because of the COVID-19. So it's very difficult to diagnostic and go to the hospital. So maybe this is the undetected of the disease of this dengue. So it's maybe the cause by why the number is decreased. So if they treated, the mortality rate is about 2 until 5%. However, if the untreated, it can reach 50%. So it's very important to detect in the first stage of the disease. So about 75% of cases are asymptomatic or mild symptoms. And symptoms are similar to other febrile illness. And many of them is small diagnosis, and there are 5 serotypes of dengue virus, and the available diagnostic test in our country is a real-time PCR. Is difficult to operate in the remote area. So our proposed solutions, we have to make the simple assay is a lateral flow. I think is already increased also in Indonesia for the diagnostic test. To develop this product, we need 2 proteins. The first is NS1 proteins as a marker or as the target, and as the detector, we need the antibody and also nanobodies. The NS1 is non-structural protein produced by dengue virus when it is entering the human body by this skin, that after the viral particle enter the cell, the NS1 is produced. So in that time, sometimes the body also can secrete the antibody. for the NS1. So the NS1 can be used as the target protein for the diagnostic, and the high level of NS1 protein were found from the day 1 and 3, making it easier for early detection. For the first, we collaborate with Dr. Widoretno from BRIN. This is for her dissertation research project, and we study how to make the diagnostic or protocol by using surface plasmon resonance. This is the scheme of the surface plasmon resonance. We can coating or immobilize the antibody, and the NS1 is here, and we can detect if there is the different, but the diffraction, it will be make the signal here. So this is our result that this SPR by immobilize the antibody specific for NS1, we can detect the existing of the NS1, and this is concentration depend. And we compare between SPR with the other method. This is the golden method, the RT-PCR, and using ELISA and literal and SPR. And we found that with the golden PCR is also same result. And we found that the baseline between the negative and positive result. So because the antibody is also difficult to produce in Indonesia, so we introduced the nanobodies. What is the nanobodies? Are antibodies with the single variable domain located on the happy chain, also shown as PHH antibodies or single domain antibodies. And because this is single antibodies, and without a happy chains, so it's very small, and it's found in camelids and the sharks, and the size also about 10 times smaller compared with the antibody. And so the advantage of the nanobodies is stable in the range of the temperature and exit pH, and can be expressed in E. coli. So for the first, we developed the recombinant of the nanobodies by transform the sequence of the protein to the E. coli, by using specific plasmid, and we succeed to transform to the E. coli, and also is expressed in the E. coli. And we found, we detect by SDS-FETCH and Western blotting that the protein expression is succeed. And also we, using the Western blotting, we found that the nanobodies is succeed to produce in the E. coli. And the last, we start to make the rapid detections by using the lateral flow, by using nanobodies as the test line, and this is for the control line, anti-MOS antibody here. And this is the early result that we found that the nanobodies also can use as the antibody for in the dot plot anti-NS1 for the rapid test in the future. And last, I would like for the conclusion that our Faculty of Engineering have to collaborate with the BRIN to achieve the impactful of research, and the collaboration between Faculty of Engineering Universitas Indonesia and BRIN could be also in academic and research sectors. Thank you very much for your attention. We hope we can collaborate, and we can contact our public relation and protocol office in Faculty Administration Center in Depok campus Universitas Indonesia. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Sahlan, for very informative presentation. Ladies and gentlemen, now are the time for the Q&A session. Please using chat box or raise your hands to give the questions to Dr. Sahlan. Any questions? Okay, if not, I have one question, Pak Sahlan. Bioengineering is must and have to be mastered by biotechnologists, also biologists, microbiologists should master the bioengineering. Because as a biodiversity country, a mega biodiversity country, we have to make a bioproduct by our own self. No other countries would like to teach us, to make a bioproduct. And then, that's a point. And what is the subject? Would you mind to tell us what is the subject in engineering, faculty of engineering and Universitas Indonesia that correlated to the bioengineering?
Okay, thank you very much, Dr. Puspita, for the questions. In the bio-related subject in our the bioprocess and environmental study program and also biomedical study program, we learn about the basics science and also the engineering science. So we learn from the biochemistry, for example, and also we have to learn for the students, learn about the principle of the basic mathematics, physics, and so on. Not only the biology, but the biology is also important, but another is also important related to the engineering basic science. And the second or the third year, we also have the student have to learn about the basic engineering, like mass transfer and separations and so on, and also related to the biotechnology and the bioprocess, like for the cell culture, fermentation, and so on. And in the last year, they also have to take some elections. For example, the election for the protein engineering and for food industry and pharmacy, and also another advanced subject. And in the engineering, we have the two streams. The first is the process streams. So how to process the bioproduct. So this is for the capstones. So the end, we have the capstone course for the product development and process development. So we have two, because in engineering, we have only two, product development and process development. So to increase the knowledge about the economy, we also have the economic engineering also in the time. So maybe this is the subject in Faculty of Engineering related to the bio. For the biomedical engineering is maybe more special because they focuses on biomedical to the medical. So it's more to the medical compared with the general biotechnology or bioprocess. So they have also specific course for the like anatomy and so on. Maybe this is the answer for your question. Thank you.
Thank you, Dr. Sahlan. Yeah, I hope we can we can accelerate our knowledge on bioengineering. Yeah, it's really important from our countries. Any questions or comments? Yeah. Maybe another questions from me, Dr. Salan. Yeah. We are the same universities, actually, in Tokyo University of Agriculture and Technology. Yeah. We BRIN have the nowadays is built good manufacture laboratories, in Cibinong Science Center, if I'm not wrong. Is it can be synergies together with bioprocess engineering in UI, and then we also have some position of a postdoctoral or visiting professor. Yeah, is it possible to work together with the facilities of BRIN?
Okay. Yeah, as I mentioned in our slide, that we can collaborate in academic and also in research. Actually, maybe for the community engagement is also, but maybe I think it's not so much. Yeah, but for the academic and for research is very very good chance for us to collaborate. For example, in the case of the bioprocess, we already have the collaborations. I think with Dr. Puspita and others. I think BRIN have a good equipment, very good equipment. So why now we also have the collaboration in the case of our research about the rapid test. So our research is in BRIN, in this case in Serpong, in the Serpong. And I think it's also some of the analytical device, is very important. I think it's also, I don't know, for the postdoctoral maybe, postdoctoral. I think it might be our postgraduate students who's already finished their study. They get postdoc or after master maybe, after master. Is it possible for the master, graduate master to not postdoc maybe as a researcher to?
We accept only for the doctoral.
Maybe for the doctoral, because now we already have, even for the doctoral program, is still combined with the chemical engineering, chemical engineering and bioprocess engineering. So the students who want to learn about the chemical engineering in bioprocess stream, so they can also enter in doctoral program in chemical engineering. Maybe I think some of the students after finish can goes to the postdoctoral in BRIN. It is possible, and I think you have the good facilities also for scale up. The scale up is still difficult in Universitas Indonesia, especially for the scale up the productions and for the another assessment like clinical study and so on. So we need the CPOB, for example, or So I think I heard that BRIN have these facilities, and we can collaborate with this. And another one for the undergraduate, because we very strong in undergraduate. For the undergraduate, I think the joint supervision is the good chance, and also for the master course. we can collaborate, and because now it's full in our laboratory, so we can have to go to another laboratory. Yeah, thank you.
Yeah, thank you very much, Dr. Sahlan. We got so many information, and then we can synergy together. As a short conclusion, the field of engineering should be learned and should be mastered by biotechnologists, microbiologists, biologists, zoologists, yeah, because you by mastering the bioengineering, we can deliver our product from the lab to industry. Yeah, thank you very much, Dr. Sahlan. Applause to Dr. Sahlan. Thank you very much. Yeah.
Thank you very much, Dr. Puspita. Maybe.
Yeah. Ladies and gentlemen, next we move to plenary session. Today we have two plenary speakers. Our first plenary speaker today is Professor Eni Sudarmonowati. Let me to read her CV briefly. Prof. Eni is a research professor at the Research Center for Genetic Engineering, BRIN. Currently also, Prof. Eni is a visiting professor at the Department of Plant Biology, ISIBio, Rutgers University, USA, awarded fund from Fulbright. Also, Prof. Eni as a member of Species Survival Commission, IUCN, Associate Editor, Malaysian Applied Biology Journal. Prof. Eni obtained PhD from School of Biological Science, University of Bath, UK in 1991, and also bachelor degree from Bogor Agriculture University, Indonesia in 1985. Prof. Eni awarded Satya Lancana Wirakarya from the President of Republik Indonesia, Best Inventor, 100 Best Indonesian Women Researchers, Ministry of Women Empowerment and Children Protection, 113 Best Innovation in Indonesia, Ministry of Research and Technology, Ministry of Research and Technology. Her research interest in genetic improvement, genetic conservation, biodiversity assessment, genetic engineering, marker-assisted selection, cryopreservation, omics, and NGS. Professor Eni will be present the material for 20 minutes. After the second plenary speaker, there will be a question and answer session for 20 minutes. Yeah. Now, allow me to welcome Prof. Eni Sudarmonowati with the topic of presentation, Genomic Approach for Modern Conservation and Genetic Improvement. Time is yours, Ibu.
Thank you, Dr. Puspita Listianti, as the moderator. Good. I think, can you hear my voice clearly?
Very clearly. Okay. Yeah.
Yeah. So, good morning, good afternoon, good evening, wherever you are. Right now in here is, what's the time now? Is 9:41 p.m. in the US. So I'm very delighted to share my talk. Just let me share screen. I think it's here. Can you see my presentations? Slide presentation? Yes.
Yes, Ibu. Clear.
Yeah. Okay. Yeah. The title is Genomic Approach for Modern Conservation and Genetic Improvement. And the outline of my talk would be, as you see on the screen, I will, as the time given to me, only 20 minutes, I will be discussing a bit quickly, but those that you want to know further details, you can refer to my presentation file, because it's already downloaded in the link that already provided by the committee. So now I move on to the first one, development of genomic-based technology. We've been facing major threat, which is global threats, and then now becoming the world crisis. Five, the main one is habitat degradation, climate change, overexploitation and use of species, invasion of alien species and pollutions, which made us facing loss of biodiversity. And also a number of animal, plant and fungi species are now listed as threatened by IUCN Red List, and most known species, like 72%, still need to be assessed. Before move on to the what the genomic can do for the conservation, I would like to share with you the progress of the development of the genomic technology since 1966, and then move on to microsatellites in 1990, and then 2000 SNPs. Everybody now use SNPs, and then for population genomics, it's more on 2010. I will be discussing more on this later on. But in here, you can see detection of inbreeding depression that it's difficult to detect using the conventional conservation technology. Just to refresh you, we have 3 sequencing generations. This is just to refresh. The first ones established in introduced in 1977 with medium width length, still low throughput, and then still high cost base. And then the second one between 2005 and 2010, already high throughput, drop the cost already. And then the third one between 2010 and 2014, it's already long read, and the price, the cost even dropped, becoming more cheaper. But then the development still go on with the establishment of NGS. There are some advantages of NGS. You can rate it here a more lower cost. And then they use a maximum number of SNPs, and also the development for genomic assisted breeding, the GWAS, the Genome Wide Association, using the utilizing the next generation sequencing, and also the GPS genotyping based sequencing, then develop a QTL map, genomic selections, and then the modification, genetic modification using CRISPR-Cas9 after the transgenic era. Just to give you a very brief information on the genomic information already available, already sequenced, and you can find it in the NCBI gene bank. You can see here all different organisms, like the most is Animalia, the green one, the most information available, and then which one is the complete sequence already, the one with the red color. So the choice is yours. I mean, depend on the objective that you targeted. So either you're going to use the whole genome sequence or the whole genome resequencing. So it really depends on the target. But the whole genome resequencing offer you the possibility to screen a neutral and functional polymorphism in high genomic resolution. And further development and moving toward functional genomic, I listed the advantages here, 7 advantages, but there are more. And here, including the wildlife and humans, and also the tolerance and quality traits assessment, and also more powerful technology or mechanism for signaling pathway. And now I would like to move on the genomic roles in conservation of endangered species. And here, why there are some endangered species difficult to be conserved, because there are some obstacles like limited mating pairs, inbreeding, and also inbreeding depressions, so that we need to evaluate which species is vulnerable or less adaptability to the local conditions. So that's why we need to use the suitable technology to address this one. So in this chart, you will see this one is more on the conservation flow chart, but the one with the red one, that actually can be addressed wider when we use genomic as compared to conservation genomics, conservation tools, and the one in green, like genetic drift, inbreeding, hybridizations, can be more precisely estimated by genomic technology. And in here, I just listed several advantages of what genomic can do for conservation, genetic conservation, such as precise genomic monitoring of changes in allelic frequency, and then quantifying the effects of genetic drift and natural selection, hybridizations, and so on. And the advantages of new approaches, not dependent on genomic information already available from the closely related species, so you can do from scratch. So for the conservations, I mentioned earlier that some endangered species are difficult to be conserved. So they declined, the population in the wild declined very rapidly. So then we need genomic approach, genomic-based approach. I mentioned some of them already earlier, but here then because it provides the potential to understand the genetic basis of interaction among species, and also genomic monitoring of changes in allelic frequency to quantify the effects of genetic drift, for example. I listed here up to 20 of the advantages to use genomics for conservation. I don't want to read all of them because of the time, but just to mention, among others, it's actually like the adaptation to local conditions, demographic history, and historical effects, size. Oh, sorry, I listed 2 in here, twice, double. And then the phylogenomics and evolutionary history, population substructure, population connectivity, and so on. And this one, because it provides, this slide shows you, because it provides quite straightforward contributions, such as addressing some problems and then offering possible solutions through genomic approaches, such as minimizing adaptation to captivity, predicting the viability of local populations. And also, it offers, with all of our roles, of course, how to slow down the bend the curve of biodiversity loss. So in here, this curve, it's actually used by UN, United Nations, on the meeting of CBD. So the down, the bottom, the gray one, the gray line, if you just do business as usual, you do nothing. And then the yellow one, increased conservation efforts, so it will bend a little bit of the curve. But the green one, if you do increase conservation effort plus more sustainable production and more sustainable consumption, then it will bend the curve of biodiversity loss. Among others, the technology is using the genomic approach because I mentioned earlier that we can prevent genetic pollution and also evaluate its conservation values and so on, and also the migration rate. So what a genomic can do for genetic improvement, because most characters actually regulated by various genes, multiple genes, complex traits, so that it's difficult and take a long time to use this conventional breeding. So using the genomic-based approach, then with the technology I mentioned earlier, the next generation sequencing, among others, and also other technology then combining with others, omics and also gene editing system, then it can fasten the way to get the targeted one. So I now move on to research and applications on conservation. So I show here the genomic-enabled discoveries and application on humans, livestock and wildlife, plants, insect, fungi, bacteria, and viruses. For example, for microbiome in health and disease, for fungi, for synthetic biology, and others for plants, for genomic selections, also for livestock, and also the diagnostic for human. This one, sorry, this one not very clear, but in here is the example in wildlife and animal, and also what the aspect also already done, already been done. For example, on wolves, monkey, birds, and also some plants also. This is just a summary. And in fish, in Amazon, in Latin America, this one, the symbol fish are Paima gigas. The genomic approach can already offer the fishing management and also genetic management for aquaculture, so that this fish that are almost extinct in the wild then can be increase the population in the wild also. And also it provides low-cost approaches and low-cost infrastructure. For Pacific salmon, this one just to give you several examples, also can give you the migration phenotypes, so that the management, conservation management can be adapted or applied using SNPs. And for Tasmanian devil, in Tasmania, this one almost extinct in the wild, but using genomic approach, the GWAS and also the SNPs, then can be prevented from extinctions, and now they can be, it's raised the population. And in bird, also in this one, the endemic and also threatened birds in New Zealand. So now using the whole genome resequencing and SNP, so they can produce this one, the conservation, the population also increase in the wild. I mentioned earlier about the adaptability and vulnerability of certain species. So this one, I just give you the case in Japan, that especially in small islands. So this one using the assessment with the whole genome sequence and also the next generation sequencing, then they can provide a recommendation for the government how to preserve this one. And also another plants on rhododendrons, also they provide the government to give effective conservation strategy. Because I'm now in the US at Rutgers University, so I know and also witness the research on this one on conservation, but as well as also for breeding of cranberry. It's famous for sourness, but it has abundant health promoting compounds. So using a bulk segregation approach with SSR and QTL, and also the GBS, the genotyping based sequencing, so they can produce they can conserve and also produce improved materials of cranberry. For woody species, sorry, not many people working on this because it's a long life cycle, but I just listed the one in Indonesia just to give example, but there are many abroad, internationally, genetic diversity, population genetic assessment and genome sequence of threatened species of Indonesian timber, such as the ironwood, the Aeschylodirexulan swageri, and also the woody species like Mangifera casturi. This is already almost extinct, but now can be propagated, and also the assessment already been done. And transgenesis and the genetic stability test. So this one, the impact of logging and the impact of forest fragmentation already analyzed, assessed using genetic markers in the past, but still the recommendations being used by the government that we need to increase the population in the wild. And this one on Ratan Manau, and also other species under Diplocapt. And now move on to the research and application on genetic improvement. So combining between gamma irradiation and microsatellite markers, two species of Acacia mangium and Paracentus falcaria can be produced, improved one, the mutant one, the straightness of the stem and the timber, and also the permanent branch, and also the height, and also the height and the branch from the surface, the branch height. And to confront with SSR, and then this one using ISSR on cassava to several traits, related to several traits, and also the SNPs for Psi gene regulating beta carotene also already been done. And also detecting the transgenic plant, this one trees on the less lignin content and high cellulase already been expressed, and this one on at least four or three species in Indonesia. One of them is endemic Indonesia, the Paracentus falcaria, and the overexpression of the genes stable after 8 years. We tested and also planted. They're still able to regenerate and still stable, confirmed with the molecular analysis. So since again, I'm at Rutgers University, I came with them, this Professor Jim Simon and Dr. Rong Di produced gene-edited basil. Basil is Indonesia like kemangi, resistant to downy mildew disease. I came with them to the experiment station there. So here, you can see the improved basil, very fresh and green. At the back, the one that I put the green light or the green pants or something, that's the wild type. It's already devastated by the downy mildew disease. So it's quite significant, the effect of the gene CRISPR gene edited basil. So this one using the gene homoserine kinase. And for rice, for sweet potato, for wheat, this one from USDA also in US. So they use a different kind of technology like GWAS, and then also other markers for the traits of grain dimension, cooking quality, beta carotene, and others like drought tolerance. So it's really depend on your target And also this one for fruit species, durian, volatile sulfur compounds, and papaya for the fruit weight, fruit length, and so on. Banana for the carotenoid rich, and other fruit for the shape and juiciness. And I listed here also the progress on functional genomics on wheat. So for tolerance and most for drought tolerance, combining transcriptomic, proteomic, and metabolomic using SNP and rapid markers. And now move on the recent and ongoing collaborations between Indonesia and in this one BRIN and Kyoto University and Tohoku University on here you can see the status endangered, critical endangered, and mostly critically endangered and endangered of 4 species using whole next generation sequencing. And also this one is just I just keep this one one of the detail of the species.
2 minutes more.
And and then now using the white durian, this is the first one. We will be the first draft genome sequence of the white durian. So this one, durian cutiensis and durian oxyanus. And then we will generate the PSMC to detect the demography structure population in the past, like 1,000 years back, and also in the future. This one a new technology as compared to PSMC, but also the MSMC, and then this one another new approach using star wave plots. Of course, there are plus and weakness, but this one does not require whole genome sequence as a reference genome. And then we're going to do another 2 white durian for producing draft genome for the first time also in the world, so hopefully it will be soon. And then for networking and collaborations, we need the all participation of all of us, governments, NGO, funding agency. And these are all different type of collaborations, bilateral, regional, global, and others. So we need to make use of that. I just give you the example in the past, a bilateral collaboration with Indonesia, Netherlands, Indonesia, UK, Indonesia, Zurich. So this one on cassava, on stock compositions, and also for extending the shelf life of cassava. And lastly, before conclusions, this one, what the research result for if is it not used by our users? So this one of the users is the government. So we need to give the recommendation for the conservation. So in these 2 books, there are also genomic approaches, the SSR and other markers that have been assessed, used to assess the conservation. And to conclude, lastly, the genomic approach provides more efficient, this one reduce time and low cost for human wellness and nature, not just human wellness, but also nature. So on the first day, yesterday, you all heard from Dr. Lily, the BCG. So this one, bioeconomy, circular economy, and green economy. And the second one, for this technology, we can provide recommendation for national conservation strategy. We can bridging the gap for breeding of elite clones or improved strains or for microorganisms and others. And also we still have a lot of homework to do because certain technology need increased or robust regeneration protocol for many species, and research need to be adopted. So that's our efforts need to be increased for that. So thank you very much for your attention. So I would be very happy to have your questions. Thank you.
Thank you very much, Prof. Annie, for introduce us your work until today. The next of plenary session is Associate Professor Dr. Insinyur Hasimi Abu Hasan Is it already here? Yes.
Assalamualaikum. Can you hear my
Waalaikumsalam warahmatullahi wabarakatuh. Yes, I can hear
voice? Is it
clearly.
clear?
Yes, very
Clear.
clear.
Okay.
Yeah.
Okay. Very much. I
Yeah,
try to
Associate Professor Dr. Insinyur Hasimi Abu Hasan is a head researcher at the Research Center for Sustainable Process Technology, University Kebangsaan Malaysia. And then also Associate Professor at the Department of Chemical Process Engineering, UKM, and also Associate Research Fellow, Research Center and Water Analysis, Faculty of Science and Technology, UKM, Managing Committee Member, International Water Association, and then obtained PhD from University Kebangsaan Malaysia. His honors and awards obtained was Anugerah Bintara Ahli Akademik Harapan, UKM, Most Valued Reviewer for Bioresource Technology Elsevier, Anugerah Bintara Ahli Akademik Harapan, also from UKM. The research interest is on a waste treatment, waste audit, resource recovery, biofilm, phytoremediation, and pycoremediation. Professor Hasimi Abu Hasan, time is yours. You will make your presentation by yourself?
yes, sure.
Okay.
Thank you, Mr.
Thank you very much.
Chairperson. Okay. I will share my screen. You can see my screen, right?
Yes,
So
we can see yours.
just a moment. Okay. 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 Thank you for inviting me to give a speech in this conference. Okay. As presented by the Mr. Chairperson, my name is Hasimi Abu Hasan from Department of Chemical and Process Engineering, and acting as head for the Research Center Sustainable Process Technology. Okay, so my topic today is on the how to treat the river or how to treat the surface water through the biological approach. Okay, just a moment. I try to remove this one. How to? Okay, never mind. Okay. So so this is my outline for the presentation, for the speech. Okay, I will start with some introduction on the environment, especially on the river. Okay, and then what kind of the river pollution in the in especially in Malaysian country. Okay, and then the treatment technologies we use to treat the river. Okay, to produce clean water. And then the challenge, the challenge to use the biological approach, and the last one, of course, we have conclusion. Okay, as introduction, as a human, we need a water, because water has an important role in our metabolic process. So this water, it will break down a large substance into a small substance. Okay, so what happens if the water contaminated by the pollutant? Okay, of course it will affect to our body. Okay, So the contaminated water will become hazardous, okay, not only to human as well as to plant, hazardous to the animal, and this kind of pollutant will expose through the drinking water. Okay. So there are a lot of pollution sources in the river. Okay. For example, from the industrial discharge, okay, the pollution from the illegal garbage. This is the common one. Pollution from the illegal garbage. Okay. And then pollution from the sewage effluent discharged to the river without proper treatment. And then from the leachate, okay, leachate landfill, and of course from the agriculture industry. Okay, so agriculture industry can pollute the river through the water runoff, okay, during the watering or during the raining season. Okay. Okay, this is some of the river water pollution. Okay, in terms of chemical compound. Okay, there are 4 categorized here. Okay, the first one is heavy metal. Okay, there are many heavy metal contamination in the river. Okay, for example, plumber, cadmium, or zinc. And then other parameter is inorganic, non-metallic. Okay, so inorganic metallic, the source for this kind of pollution is from the sewage industry, from the industrial industry. Okay, and for example, of the inorganic non-metallic pollutants, nitrogen compounds, sulfate, or chloride. Okay, and then what else? The river water pollution. Okay, endocrine disrupting chemical, EDC. Okay, and disinfection byproduct. Okay, these 2 categories are under micropollutant types. Okay, we could not see by our naked eye. We can analyze both categories using high-end equipment and not very easy to analyze. So this is the current issue in the river, okay, around the world. Okay. So and then other kind of the water pollution are physical. Okay. So the physical contamination contribute by the garbage. Okay. And then once we throw the garbage into the river, so it will contaminate the river, of course, as well as the color. It will create the bad color, the black color, the brown color of the river. And then we can see the turbidity, the clearness of the river become reduced, okay, through the turbidity measurement. Okay, this is some example, the water pollution in Malaysia. Okay, some news, okay, about the contamination. Okay, and that news in Malay, I think you can understand. Okay, you can see the fish in the river. Okay, died because of the pollutant. The river water become toxic. Okay, and not only die, some aquatic plants also become yellowish, start to decay. Okay, and some other example. Okay, in Malaysia, there are many. Each year we can hear about the water contamination, not only in Malaysia, also in other countries, especially in developing countries. Okay, this is some other example. So you can see this in English. The river water turn green. Okay, this is because of the dye discharge. Okay, from the textile industry. So they discharge the dye directly to the river without any proper treatment. Okay, so in Malaysia, we have a conventional treatment, okay, to treat the river water. if the river are very contaminated at high level, of course, the conventional treatment could not be operated for the production of clean water. Okay, so in Malaysia we have the treatment of the grid chamber. Okay, this one, and bench stream. This is to remove the garbage. Okay, or to remove the large particle. Okay, for example, the dead leaf, the trunk of the plant. Okay, and then we have the aerator here. Okay, aerator is purposely to reduce the light heavy metal, for example, the ferrum, the manganese. Okay, and then followed by the flocculation and sedimentation tank. Okay, this is to remove the dissolved organic material. Okay, for example, to reduce the suspended solid, to reduce the dissolved metal, so we use the flocculation process. And then we have a sand filter, okay, before the end of the treatment. Okay, sand filter to remove residual suspended solid. And the last process, we treat the water by the chlorination. This is to ensure there is no microorganism contamination in the water. Okay, so our purpose, okay, why we develop a technology to treat the river water using the biological approach is in the current technology we could not remove the nitrogenous contamination. For example, we could not remove the current technology could not remove the ammonia, could not remove the nitrate, could not remove the nitrate. So by having the biological approach, okay, so those pollutants could be removed effectively, and at the same time the water treatment in Malaysia continuously operated, okay, without any shutdown. So this is what we propose and what we study in Malaysia in my department. Okay, biological treatment is one of the advanced treatment. Okay, so it started popular since 1900, but not for the water treatment. It's popular for the wastewater treatment. Okay, so the application is still limited around the world. Okay, but for the developed country such as US, Canada, they have very well knowledge on the technology for the drinking water production or for the river treatment. Okay, so of course, when we're talking about the biological approach, we have to make sure that the process use non-pathogenic bacteria. Okay, so this kind of bacteria will act as biocatalyst for the biochemical oxidation, and then it can degrade the pollutant in the river. So the technology could be proposed before the chlorination process. Okay, because if you propose at the end of the process, it might be contaminated with the microorganism. But if you propose before the chlorination process, so the microorganism contamination could be avoided. Okay, so now we go to the what kind of the technology for the biological approach. Okay, so we can use the slow and rapid sand filtration. Okay, so we have published a few articles on this technology, and this technology could remove the light heavy metal. For example, it could remove the manganese, it could remove the ferrum, it also could remove the plumbum, okay, in the river water. Okay, so but the lacking of this technology, it could not be operated if the heavy metal too high in the river. Okay, too high means it could not exceed 2 ppm. Okay, if the concentration exceeds 2 ppm, so we could not use this technology. Okay, because the heavy metal could provide a toxicity to the microorganism. Okay, so this is some explanation about the sand filtration. Okay, there are two types of sand filtration. The first one we call slow sand filtration, and second one is rapid sand filtration. Okay, so this process used normally we used to filter out the suspended solid and light heavy metal. Okay, so the sand filter, okay, that fail in the reactor, okay, will act as a support for the biofilm growth. Okay, once the biofilm grows, it can provide a space for the biological activity. Okay, and we can design the sand filtration in a column shape or in a rectangular shape. Okay, this is the biofilm layer on the sand surface. This is in German language. Okay, we call it smart Dutch. Okay, and this biofilm layer contain extra polymeric substance. Okay, so this substance could react with the pollutant through the bioaccumulation, through the bioabsorption, and of course we can produce the clean water. And this is some of the performance for the biosand filter to clean the river. Okay, especially the ferro and manganese as a light heavy metal, as well as the ammonia. Okay, we can also treat the ammonia if the ammonia level high, more than 3 ppm, so we can use the sand biofilter. Okay, next is biological activated carbon. Okay,
You have 5 minutes more, Sensei. Yeah.
Okay,
Thank you.
I will speed up my presentation. Next is biological activated carbon. So this kind of technology use the activated carbon to absorb the pollutant. Okay, so there are two mechanisms for the biological activated carbon. The first one is aerobic process, and the second one anaerobic process. So the activated carbon or the adsorbent could be regenerated through the microorganism role. Okay, so the microorganism will consume the pollutant, and at the same time the pollutant from the activated carbon will consume back by the microorganism for their growth. Okay, this is the performance. It can reduce the micropollutant. Okay, something like EDC. Okay, and then trickling filter, another treatment. Okay, this is not a common technology for the trickling filter, but we can use the trickling filter to remove the manganese, okay, iron, and also ammonia. Of course, this technology will involve the microorganism, something like manganese oxidizing bacteria, ammonia oxidizing bacteria, for example, Nitrosomonas, Nitrobacter. Okay, and also we can use the biological aerated filter. We have published more article for this kind of technology. Okay, this will involve the using of floating carrier. Okay, the floating carrier will act as a house for the bacteria to grow, to attach, and at the same time it can provide a large surface area for the biofilm, for the contaminant removal. Okay, this is the performance. And then we also could use membrane bioreactor. This technology combination of the bacteria, especially bacteria, and the membrane. Okay, and there are two type membrane technology. Okay, we call it as attached growth membrane bioreactor, and we can also set as ion exchange membrane bioreactor. Okay, so normally we use the membrane bioreactor at the end of the process before the chlorination. Okay, And also we can use the moving bed by film reactor. Okay, this technology combining the activated sludge process and also with the aeration, we provide some aeration for the bacteria. Okay, and the aeration could create the dissolved oxygen concentration in the contaminated water, okay, normally at 2 until 5 ppm. Okay, so there are also some integrated biological approach. Okay, with other technology. Okay, for example, we integrate the membrane. Okay, membrane bioreactor with the coagulation process. We add some chemical before the treatment. Okay, so the chemical will reduce the suspended solid or will reduce the light heavy metal, and the treated water will go to the membrane and it can increase more the pollutant removal. Okay, and then we also integrate with the absorption technique, okay, between the absorption and the membrane. Okay, we call it as biological activated carbon membrane bioreactor. Okay, so this is the combination. And then what is the challenge in using biological for the drinking water treatment? So we have to face, we have to solve how to make sure the treated water are free from the pathogenic microorganism contamination. Okay, this is the challenge. We should have a frequent monitoring on this parameter. And then the selectivity of the microorganism towards the pollutants. Okay, not all microorganisms have capability to remove the pollutant. So we should provide the system with a good microorganism that could remove what parameter we need. Okay, and then the third challenge is to maintain the growth of the microorganism in the system. Okay, this is very challenging, and of course, once we produce the drinking water or we treat the river, what is the consumer perspective? Okay, are they worried about the contamination of the microorganism or not? Okay, so we have to do some survey for this. Okay. And at the end, for the conclusion, of course, this is a new technology or our future technology for the river water contamination, as well as especially for the developing country. And there are many area that we can explore. And of course, this technology could be growth well in developing country. Okay, thank you. I think that's all my presentation. Before I end, I want to say something. The earth is beautiful. Let us maintain the beauty by caring for the environment. Okay. Okay. Thank you very much, Mr. Chairperson.
Thank you very much, Dr. Hasimi Abu Hassan. Ladies and gentlemen, now are the time of Q&A session. Please using chat box, chat feature of the Zoom and raise your hands, and then from whom the questions will be delivered. We have 10 minutes for the question and answer session. And then there is a one question from the chat book to Prof. Annie. According to your experience, what method is the most effective and efficient in transferring the desirable gene into the plant? From Ibu Elok, BRIN.
Okay, thank you for the questions, Ibu Elok. Yeah, so nowadays, because of the development of CRISPR-Cas9, this is the most effective way. I mean, efficient, because efficient when we talk about the resources. So it's quicker in terms of time. I mentioned in my slides, I showed to you all the successful research using CRISPR-Cas9 on basal at Rutgers University in the US. So this one, they only needed 2 years to get the gene-edited plants as compared to the one that using the conventional breeder, they need 7 years. And then how to deliver the gene after it's cut, by CRISPR-Cas9. For me, for my experience also, and also when I'm here at Rutgers, using particle gun as compared to agrobacterium tumefaciens, because when you use agrobacterium, it's almost most of the time you will be like deleting or to get rid of the bacterium from the plants. I mean, if the concentration is too high, then your time is consumed for transferring or get rid of the agrobacterium. But using particle gun bombardment is better, because then you don't have to be disturbed with the agrobacterium. So in here at Rutgers University also, they use the particle gun bombardment of BioRad. I mean, last time I used not BioRad because we used the cheaper one, but less effective. So I think we need to have the BioRad also. And also, although either you use the conventional genetic engineering or using the transgenesis or CRISPR-Cas9, again, as I mentioned in my conclusions, one of the list that the regeneration procedures is need to be established. So no matter how high or how advanced is your technology, if you want to improve genetically, you have to establish the regeneration system. Yeah, so that's my answer. I hope that's satisfy you. Thanks.
Okay. Thank you very much, Prof. Annie. And then from the chat book to Prof. Hasimi from Ibu Penny Lestari. The result of this research are very good and protecting the earth from contaminants. Is the use of bacteria of microbes to treat pollutant also safe for human? Or how to manage these microbes? How far have the results of this research progressed? Has it been applied in product form? How to get it? Please, okay. Dr. Hasimi. Yeah.
Okay, thank you for the question. Okay, apologize. Okay, there are few questions, right? Okay, how to manage this microbe? Okay, during the treatment, okay, we could monitor the growth of the microbe, okay, through the what we call it mixed liquor volatile suspended solid, okay. Or we can measure through the adenine triphosphate analysis, okay, ATP. So this value represent the growth of the microorganism. Okay, once we produce the treated water, okay, after the biological treatment, we will treat with the chlorination. Okay, so the chlorination will destroy all the microorganism, and we will make sure after the chlorination process, there is no microbe contamination in the water. Okay, this is what we have done. Okay, and then so of course the safety of the treated water to the human. Okay, so this is the challenge that we have faced. Okay, so the consumer will worried to consume this water because they think it contaminated with the microorganism. Okay, so we have to convince the consumer or the human that the water is safe. Okay, because we are using the non-pathogenic bacteria. Okay, not pathogenic bacteria. Okay, so and we also control with the chlorination. Okay, before we distribute the water to the human. So through this control, okay, so we can ensure that the water are safe to the human. Okay, so and then so far this project is still under research progress. Okay, not apply in a real scale or pilot scale. Okay, of course there are more study to be explored. Okay, especially the consumer perspective. Okay, and there are one question about the economic. Okay, so for the economic test or analysis, this still ongoing. Okay, but at the lab scale, not at the pilot or real scale. Okay. Okay.
Yeah.
That's my answer.
Okay, and I want more answer. How about the study from economic side when it will be applied?
Yeah, of course, in terms of economic side, okay, from the review, okay, this technology should be more cheaper compared to other chemical technology. Okay, because we use less chemical. Okay, and then it easy to operate. Okay, easy to operate. That doesn't mean we do not need a competent staff or competent operator to operate. Okay, still need the competent operator, but easy to operate, easy to learn. Okay, just to competent how to manage the microorganism growth in the system, and of course, economic side is more cheaper compared to other technology. Okay.
Okay, thank you very much, Dr. Hasimi. We will be finished for this plenary session. Thank you very much, Prof.
Yes,
Any.
there are two. Sorry, there are two.
Oh yeah.
There are two raising hand.
Oh, I'm sorry. Oh yes.
Yeah.
Pak Panji and Ibu Yeni, please. First, Pak Panji.
Do I still have time, by the way?
Yes, we still have 1 and 2 minutes.
All right.
Okay.
All right. I'm just going to make it quick. I have one question for Bu Eni and one question for Pak Hasimi. For Bu Eni, I am studying microbiome now, and I learned that the loss of the microbiome would influence the loss of the high organisms as well. Do you think that targeting the microbiome in addition to the genetic identity of the microorganism would help to preserve the endangered species? And for Pak Hasimi, I am now doing a project with KU Leuven about the use of microbial inoculant and as well as the horizontal gene transfer in microbiome to reduce the pollutant. And one of the key challenges that we face is to maintain the survivability of the microbial inoculant. How did you maintain yours in your system? Thank you very much.
Thank you very much. Please give
Can
the
I
short
answer?
answer.
Yeah, very short one. Okay, before I also short because of the time. Yes, definitely studying the microbiome will affect the degrading or increasing the populations or enhance the loss of biodiversity or maybe improve the population size. That's why the study in microbiome is very important, and also because it's also related to the gut, even in the intestine of the animal or wildlife or even human. So that need to be studied further. Yeah, so that's why the study in microbiome is very important, because it's related to the extinctions or the survival of the species. Yeah, that's my quick answer.
Ibu Eni. Dr. Hasimi, please. Yeah.
Okay. Okay. How to maintain the growth of the microbe in the reactor? Okay. What we do is, the first step is we have to make sure the incoming river water to the system is not at very high contamination. Okay, so we should remember this technology is used not at the first stage. We could use at the second stage or the third stage. Okay, if the concentration of the contaminant is very high, so it will kill or will become toxic to the microorganism. Okay, the contamination depends on what parameter we want to treat. Okay, and for example, if we want to treat the ammonia, okay, we have to make sure the ammonia level after the first stage of treatment, it should be not more than 3 ppm. Okay, so we are talking about the water, so the contamination should be not high, and from that it will maintain the growth of the microorganism, and we can monitor the growth through the mixed liquor, volatile, suspended solid. Okay, that is what we have done in our research.
So by that you mean you get the microbiome to adapt to the environmental stressor that would...
Yes.
Okay. Okay.
Yes,
right.
we have to do that. Okay, to make sure it can adapt with the environment. It can have a good removable stability performance.
All right. Okay. Thank you very much, both.
Okay. Okay.
Thank you very much. I'm sorry, Ibu Yeni. The time is up, and then thank you so much again, Prof. Eni, and then Dr. Hasimi, for the very informative and then inspiring presentation. Yeah, I move this session to MC.
Thank you.
Yeah, thank you so much.
Thank you.
Thank you, Prof. Eni. Thank you, Dr. Hasimi, and also thank you, Dr. Puspitalis Dianti, as the moderator of the sessions. Ladies and gentlemen, following to the next agenda, we will have the parallel sessions consisting of 5 rooms with different topics, and also each of room will be led by moderators. So the first room will be bioresources and biodiversity. The second room will be nutraceuticals and natural products. The room 3 will be biotechnology and bioinformatics. Room 4 will be medical sciences, and the last room, biorefineries and circular economy. So please kindly choose the room based on your interest by clicking the breakout room feature at your Zoom. If you haven't found it yet, you can click more with 3 dots at the corner of the left of your Zoom meeting, then join breakout room there. The room 1 until 5 are available there. Once again, please kindly choose the breakout room by clicking feature more and then join breakout room. Allow me to read once again the room. Room 1 will be bioresources and biodiversity. Room 2, nutraceuticals and natural products. Room 3, biotechnology and bioinformatics. Room 4, medical sciences, and room 5, biorefineries and circular economy. As you can see on your screen right now,
Okay, could you hear my voice clearly? Okay, we will wait until all participants and presenters joining in this room. All right, shall we begin this parallel session? Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome and thank you for joining room 3 in session 1. I am Fatima. I will be your moderator for this session with a topic about biotechnology and bioinformatics. So I would like to invite our 6 presenters in this room. Pak Tulus Maulana, are you already available? Okay, Pak Win Kusharyoto. Pak Win, already here? I think not yet. Okay. We'll move to other. Miss Hairunisa. I think this still not available. And about Pak Sutikno?
Present. I'm here.
Okay, Pak Sutikno. Pak Rikno? Oh yes, already here. And Miss Eva Agustriana? Oh, I think we should wait another 1 more minute for Pak Win Kusharyoto and Hairunisa. Hairunisa is already here. Pak Win? Oh, not yet. Okay, for Pak Win, we put in the last, and then we pursue with the one that already joined with us here. All right, before we begin, I would like to deliver the order during this parallel session. For presentation, our technical team will display the recorded video presentation of each presenter. And then after recorded presentation of all presenters is delivered, we will open the Q&A. You could raise your hand or write down your question in the chat box. Hopefully, we will end this session by 12:10. And just a little note for all presenters, we will give evaluation for nomination of the best oral presenter. So please prepare for yourself. All right, let's move on to video presentation. Please welcome our video presentation from Pak Tulus Maulana. Screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you for opportunity. Thank you for moderator for this opportunity. Today, I would like to present our research paper with the title Mitochondrial Membrane Potential of Barley Bull Frozen Towards Semen, assessed by mitostatus TMRA. For introduction, the use of flow cytometry for sperm quality assessment is the current solution for rapid and precisely reproducible assessment of cell and thus becoming important in the field of veterinary. Numerous fluorochrome with well-defined excitation and emission spectra have been described for the evaluation of sperm functional status. Example of sperm characteristic that can be measured with flow cytometry include plasma membrane, acrosome integrity, mitochondrial status, chromatin integrity, oxidative stress, and intracellular level. The mitochondrion is gaining more attention as a biomarker of sperm health and fertility as a result of recent studies on the psychology of human and animal sperm. Sperm mitochondrial activity is studied and utilized as a metric for spermatozoa viability in vitro for evaluation of their ability to fertilize and measurement of the efficiency various sperm diluent. The mitochondria are an important source of energy for the motility of sperm, and alteration in the mitochondria may be used as a quality indicator of semen. The aim study of this research was to examine mitochondrial membrane potential of spermatozoa by the applicability of novel fluorescent probe mitostatus TMRA by using flow cytometry and correlation with the motility pattern of candidate and certified bull frozen semen. For material and method, we use 4 candidate bulls, Bali bulls, and 4 certified Bali bulls. The frozen semen we collect, we produce from 8 Bali bulls, and we evaluate from a 3 batch production. And then the frozen semen with thawing in water bath for 30 seconds in 37 degrees, and then we analyze using the computer-assisted semen analyzer or CASA, sperm vision Minuted Germany, with the parameter progressive motility, hyperactive, linear, and curvilinear. And then for the mitochondrial membrane potential assessment, we use frozen semen, and then with thawing. Then we add the mitostatus TMRA for 200 nanomolar, and then we incubate for 30 minutes. And then we analyze using flow cytometry, BD accuracy, and NM+. And then we use unstained as the control. For result and discussion in table 1, motility pattern, progressive motility and hyperactive, and mitochondrial membrane potential in candidate and certified bulls. In table 1, you can see in this study, so there were no significant difference for the progressive motility, linear and curvilinear parameter in candidate and certified bull. But on the hyperactive parameter, there is a significant difference with p-value less than 0.05. Pardede et al. Reported that the spermatozoa with progressive motility more than 47% were able to produce a conception rate of more than 65%. And another paper reported that the motility and movement pattern values of spermatozoa indicated the presence of metabolic activity and structural integrity of mitochondria and spermatozoa flagella. And then we can see the percentage of MMP parameter was 19.8.25 and 19.7.89 respectively. That is so there is no significant difference between candidate and certified bull. Previous study reported that semen quality parameters such as mitochondrial function were associated with fertility. And then in table 2, we can see the correlation mitochondrial membrane potential with motility pattern. The Pearson correlation between MMP and L motility pattern was negative, with p-value more than a significant level, p less than 0.05, which indicated that as MMP increased, motility pattern parameter were decreased. Espinosa et al. Reported that a decrease in spermatozoa motility related to mitochondrial activity, mitochondrial damage, result in impaired production of adenosine triphosphate or ATP. In figure 1, we can see the picture forward scatter and side scatter like data were collected. This is we getting the population that we want to measure or we want to analyze, and for the B picture, it means the unstained or the control group, and the C is the staining with metastatus. In the staining in the control group, there is a high, there is a peak, but in the left side, but in the staining area is the left in the right side. We can see in the picture, figure 1 in D picture, non-apoptotic of cell with the polarized mitochondria will glow red, and the apoptotic or depolarized mitochondria will have reduced red level or will dark red. In the study, metastatus type MA can be used to assess apoptosis mitochondrial membrane potential and mitochondrial depolarization. Non-apoptotic or cell with polarized mitochondria will glow red in figure 1. And based on the study, the combination of flow cytometric and computer assessment and laser assessment provide a rapid and objective measurement for sperm quality analysis.
Okay.
For the conclusion, metastatus TMRA, it can be used to be a marker from frozen towel bovine semen mitochondrial membrane status in both candidate and certified volleyball. Maybe that is our presentation. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Tulus, for interesting research and nice presentation. And then we continue for next video presentation from Pak Wins Kusaryoto. Okay, screen is yours.
Hi everybody, Assalamualaikum warahmatullahi wabarakatuh. Today I would like to present our work in subcloning, expression, and molecular characterization of L-arabinose isomerase from Thermotoga termarum. This work was done by two of our students, Nida and Fitra from Surya University. L-arabinose can catalyze the isomerization of L-arabinose to L-ribulose, but the enzyme is also capable in converting the galactose and the tagatose. This is interesting because the tagatose is an isomer of L-fructose and has around 92% sweetness as sugar. Thus, the tagatose can be used as a low-calorie sweetener that demonstrate an anti-hyperglycemic effect, which is of advantage for example, diabetes patient. The objective of the study was to subclone the ARA-A gene from Thermotoga termarum into the so-called P-ray expression vector to express the protein in E. coli and also to purify the enzyme by immobilized metal affinity chromatography. We also generated a 3D structural model for this enzyme with the bound substrate, the galactose, and evaluated the catalytic set of this enzyme. For the isolation of the enzyme, we use the genomic DNA of Thermotoga termarum DSM5069 we purchased from DSMZ in Braunschweig, Germany. We subclone the gene into the PTA2 cloning factor from Toyobo. For the isolation, we use the primers, which contain the restriction site for the transition enzyme NDE1 and EXO1, and also a sequence encoding the C-terminal histag for the purification of the protein. After cloning into PTA2, we perform restriction analysis by using both restriction site, NDE1 and EXO1, and also DNA sequencing just to verify that the inserted ARA-A gene was exactly the gene we wanted to have. And the results show that the gene was successfully isolated from the genomic DNA of Thermothoca temarum. The gene has the size of around 1,500 base pairs, and the gene was also corrected into the PTA2 cloning factor. After the regression analysis, the results show that the size of the PTA2 and also the gene was correct. And at the same time, we perform digestion to obtain the pire vector using the same restriction enzyme. And in this case, the restriction enzyme has around, the clonate vector has around 4,200 base pairs of size. After isolation and purification of the gene and the pire vector by using Quick gel extraction kit, the gene and the expression vector was ligated using DNA ligase, transformed into E. coli top 10, and we name the expression vector pire RAE TTAI. And after isolation of the positive E. coli clones, the recombinant vector were verified by regression analysis using NDE1 and X1, as well as DNA sequencing, just to make sure that the gene was inserted in frame into the expression vector. This is the construct of our expression vector. This expression vector has RAB promoter, which can be induced by L-rhamnose to produce the protein. In this case, L-arabinose isomerase from Thermothoca temarum. The protein has a his tag at the C terminal. And the result of the subcloning into the pire vector show that the gene was correctly inserted into the vector, and the regression analysis show that the result can be confirmed by the method. We also perform DNA sequencing just to make sure that the protein, the gene was correctly in frame inserted into the expression vector. We perform multiple sequence alignment just to know the relationship or the similarity of this arabinose isomerase from Thermothoca temarum with other arabinose isomerase from other organisms, and compared to Thermothoca neapolitana and Thermothoca maritima, the LRE from Thermothoca temarum has identities of around 80.6 or 80%. Compared to Q-Bacillus stearothermophilus and Q-Bacillus castrophilus, the identity was lower, 62.9% and 62.1%. Even if you compare the similarity with the Lactobacillus brevis, the identity was only 46.8%. The protein was expressed by E. coli NICO21 in the cytoplasm as a C-terminal his tag protein. The expression level was the highest if we induce the expression using 100 micromolar of L-rhamnose. And after isolation of the protein from the cytoplasm of E. coli, the protein was then further purified by IMAC on Talon TM matrix. This Talon matrix has the color of pink, as you can see in the picture, and the elution of the protein can be done by addition of imidazole into the buffer solution. The protein can be expressed and purified successfully using IMAC, and it has around 56 kilodalton in size, which correspond well with the calculated molecular weight based on the amino acid sequence of the protein. And you can see here a faint band just below the band for arabinose isomerase. This band may correspond to the histidine-rich proteins like D, CBD, commonly co-purified by IMAC if you use E. coli NICO21. We also perform homology modeling and substrate docking using Swiss model workspace based on the available structure of arabinose isomerase with the bound cation manganese 2+ from Q-Bacillus castrophilus. The model was further verified by project Verify 3D and QMIN. After that, we perform a docking experiment using Autodoc Vina And in this case, the substrate, the galactose, was placed into the active site of the enzyme. And during the docking experiment, the substrate and the amino acid residues around the substrate were said to be flexible. And the results show that the substrate, the galactose, was bound by similar amino acids previously identified responsible for the binding of the substrate in the catalytic site of the enzyme. For example, you see here two hydrogen bond between glutamic acid in the position of 300 and glutamic acid in the position of 328 with the bound substrate, and also other residue responsible for the binding and recognition of the substrate can be verified. In conclusion, we have successfully subcloned, expressed, and purified the recombinant L-arabinose from Thermotocatemarum in E. coli cytoplasm. And we also built a 3D model for this arabinose isomerase for further modification of the enzyme. The further modification may include, for example, the enhancement in enzyme activity, the change in specificity from L-arabinose to the galactose, for example, and also in a modification of the stability of the enzyme. For the experiment is also still necessary to optimize the expression level of this enzyme by E. coli fermentation. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Win, for an inspiring research and great presentation. We move to next video presentation from Ms. Hairunisa. The screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for giving me the opportunity to present my work with the title of Image-Based Growth Evaluation and Potassium-Sodium Ratio of Transgenic Rice Lines Harboring ATAVP1 Gene Under Salinity Stress. So the background of this study is soil salinity, which is a serious and global threat, especially for agriculture. There are some main causes of soil salinity. It can be because of saltwater intrusion, harsh climate, uneven rainfall, wind or rain-borne salt deposition, and can also be because of human activities such as irrigation, the application of inorganic fertilizers, and soil amendments. Now, these two pictures showing the difference and the devastating effect of soil salinity. And based on the data, these are the top 8 countries with more than 1 million hectares of area under saline water irrigation, and the top 4 being Pakistan, China, United States, and India. So looking at the situation, the ACPFG, in collaboration with FIBA Agritech India, developed transgenic rice lines carrying ATAVP1 gene to tackle soil salinity issue, particularly in India. So what is ATAVP1? So AT stands for Arabidopsis thaliana, and AVP1 stands for vacuolar hydrogen PPases, which are known in plants for their role in facilitating the sequestration of sodium ion into vacuoles. And there are many publications showing that under saline conditions, the constitutive expression of AVP1 increased the shoot growth of Arabidopsis, alfalfa, barley, rice, creeping bentgrass, cotton, and peanuts. And this picture is taken from Schilling et al. 2014, showing transgenic barley expressing AVP1 had increased shoot biomass and grain yield compared with the wild-type plants in a field with high salinity. So based on what they know about ATAVP1, the ACPFG then developed 2 transgenic rice lines, which are the IR 411-17-10-2 and the IR 411-19-61. And these transgenic rice lines are homozygous. So we set up experiment to evaluate its performance under salinity stress. So we use these two transgenic rise lines, and we also incorporated two controls, the IR64 as the background genome and the null segregant. We measure two parameters, which are the growth or the biomass and the potassium-sodium ratio. As for the method, we started off by sowing the seeds for 7 days, and after 7 days, we transplant the seedlings into pots, 3 plants per pot, and we grow them for 10 days. And after 10 days, we pick one best plant to undergo image-based phenotyping. And this is the chosen plant. We grow them for another 14 days before we divided them into 2 groups. The first one, or the first group, is the salt-treated group, was given 120 millimolar of sodium chloride, and the second group is the control group, was given a normal watering condition. So how does this work, the image-based phenotyping? So the image-based phenotyping was done for 28 days, and during that day, every morning, this conveyor belt bring the plants into the imaging chamber. In this imaging chamber, 3 cameras are capturing the image of the plants, from 2 from the sides and 1 from the top. This is the illustration of how the cameras are working, taking images from the sides and from the top. And to maintain each of the plants in the same condition during the experiment, every day we also weighed the pot to figure out how much water had evaporated, and we replace the water by adding the water through this hose to the final target weight. And these are the results that we get from the software by extracting the data from the software. So these are the growth curves for IR411-17-10-2, and these are the growth curves for IR411-1961. The y-axis is the projected shoot area in kilopixels, and the x-axis is the days after planting. And these colored lines represent the individual growth of the plant, and this solid line represents the average growth of the transgenic. The dashed line represents the average growth of the IR64, and the dotted line represents the average growth of the null. If we are just looking from this curve, we get an indication that the IR411-17-10-2 performed better than the IR411-1961. We can see that here, even under normal condition, the growth of the second transgenic lines were worse than the IR64 and the null. And this is the projected shoot area on the last day of the experiment. We want to extract the data because it can be used to represent the projected biomass, and we want to see if any of the transgenic lines accumulates higher biomass than the controls. However, statistical analysis showed that there are no difference in biomass accumulation between the transgenic lines and both controls. In fact, this particular transgenic line, which is the second line, performed poorer than the 2 controls, the IR64 and the null. Another parameter that we measured is the leaf potassium-sodium ratio. So on the day of salt application, we marked the 5th leaf by using black marker. And then on the last day of the experiment, we take that marked leaf and then we measure the content or the concentration of potassium and sodium ions by using flame photometer. Now, these are the results. And from the chart here, we can see that statistically there is no significant difference of potassium-sodium ratio between all 4 lines, either under control or salt condition. And from this chart, we can also see that there is a significant drop of potassium-sodium ratio when the plants are subjected to salt stress. This implies that a large number of sodium ions enter the plant cells during the salt stress. So for conclusion, the first one is the projected biomass and the potassium-sodium ratio of 2 homozygous transgenic rice lines harboring ATAVV1 were not significantly different with the non-transgenic IR64 and the null control, both under salt and normal conditions. And by evaluating the overall growth and the potassium-sodium ratio, we found no indication that overexpression of ATAVV1 gene enhanced salinity tolerance in the 2 transgenic rice lines at vegetative stage. And as a suggestion, further studies should be done to evaluate the ATAVV1 gene expression in the 2 transgenic lines and to observe the plant's performance at generative stage, including its yield. Thank you.
Thank you, Ms. Nista, for an interesting research and nice presentation. Then we continue for next video presentation from Pak Sutikno. Screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thanks for the opportunity. Let me introduce myself. My name is Sutikno. Today I will present the result of our research entitled New SNP in ADG1 gene, 5 UTR, and their association to barley cattle meat quality trait. Okay, next. And this is introduction. As we know, barley cattle, Bos javanicus, are indigenous cattle from Indonesia, which have good adaptation in harsh environment, have high production trait with 45 to 57% carcass percentage, and have good reproduction trait, 70 to 90% service per consumption. We know the barley cattle has potential to be developed to premium beef cattle in Indonesia. This is editorial differentiation sphingolipid G protein coproreceptor 1 gene. As we know, ADG1 gene, this gene has potential candidate for selection for beef cattle because this gene affected to adipogenesis and intramuscular fat. Adipogenesis in intramuscular fat were affected to marbling score in cattle. Some literature say the AG1 gene is associated with intramuscular fat deposition in Japanese black cattle from Japan and Hanwoo cattle in Korea. But we don't know yet in barley cattle, so we conduct this research material and method. For polymorphism analysis, we use 88 blood sample, including 30 blood sample from barley cattle, 20 blood sample from Ongol grade cattle, 20 blood sample from Brahman cattle, and 18 blood sample from Limousin. For association analysis, we use 30 barley cattle, age 12 to 15 months, 17 steer, and 13 heifer. DNA extraction was performed using Kiagen DNA extraction kit procedure, amplification using thermal cycle AB system equipment. For sequencing, we sent to fish-based laboratory. For amplification, we use primer, show in slide, where we design ourselves. And for PCR condition, you can see in the slide 2, we use annealing 60 degrees Celsius in 30 seconds, and we use 35 cycling for PCR condition. And for mid-quality thread measurement based on ultrasound parameter, we check is backpack thickness, longest dorsal thickness, ram thickness, marbling score, and intramuscular fat percentage. Characteristic of mid were predicted using ultrasonic linear transducer. Okay, this is the result of our research. The ADG1 gene was successfully amplified at temperature 60 degrees Celsius for 30 seconds. The PCR product was 411 base pair. In belly cattle, we found 3 novel SNP. And the name SNP is C-399. C changes to T. Second SNP is C-326. C changes G. And third SNP C-273. C changes to G. And that 3 SNP were polymorphic in belly cattle, but monomorphic in ongol grade, Brahman, Elimusin cattle. While the common SNP C-312A change to G were reported in Japanese cattle and Hanwoo cattle, was monomorphic in belly cattle. And this is table of genetic frequency, allele frequency for belly cattle and other cattle. We find in belly cattle 3 SNP, this face SNP. We just find 2 genotype, CC and CT, low heterozygositas, but still in Van de Wembeck equilibrium. Second SNP, same, we just find 2 genotype, CC and CG. While in third SNP, we found 3 genotype, CC, CG, and GG. This is a common SNP in Japanese black and Hanwoo cattle, was monomorphic in belly cattle. But there is polymorphic in Brahman and Limousin cattle with low heterozygosity. Just find 2 SNP, AA and AG. And this table of association of the novel SNP with mid-quality thread in belly cattle. For first SNP, we just find 2 genotype and we associate it. The result is not significant associating. Second SNP, same. We just find 2 SNP, 2 genotype and not associate with any parameter. While for third SNP, we find 3 genotype, but unfortunately, GG genotype we just find one sample. So the result not significant too. Maybe it can be significant if we find 2 or 3 sample in here. Maybe this can be significant on this parameter, but in this research we didn't find it. Okay, in this research we can conclusion we find 3 polymorphic SNP in Bali cattle, but monomorphic in Ongol grade, Limousin, and Brahman cattle. None of these SNP were associated with some mid quality parameters. For future study, maybe we use more sample to get associating this SNP. Thank you for the attention.
Thank you, Pak Sutikno, for curious research and gentle presentation. And then we move to next video presentation from Pak Rikno Harmoko. The screen is yours.
Ladies and gentlemen, good morning. My name is Rikno Harmoko. I am from Research Center for Biotechnology, National Research and Innovation Agency, BRIN. Today, I would like to present my work regarding to the functional studies of Arabidopsis thaliana ascorbate peroxidase 2 gene in nickel stress tolerance. Heavy metal pollution is a serious problem for environments, agricultural land, and human health. Accumulation of heavy metal in soil and water can contaminate feed, food, and affecting human health. Accumulation of heavy metal in soil and water is due to anthropogenic activities or human activities such as industry, agriculture activities, and mining. Some heavy metal like copper, nickel, and iron or zinc are essential micronutrient for plant, but excess accumulation of this essential metal in soil led to the plant toxicity. In addition, low concentration of non-essential metal like CD disturb biochemical process and decrease plant productivity. Absorption of heavy metal by plant can lead to the photosynthesis reduction and reduction of chlorophyll accumulation, growth inhibition, and accumulation of oxygen, reactive oxygen species or ROS. How metal brings stress to the plant? Redox active metal like iron, copper, cobalt can enter Fenton-Haber reaction, in which it can accumulation of superoxide. Non-redox active metal like nickel, cadmium can inhibit cellular antioxidant enzyme like superoxide dismutase. So indirectly, it can contribute to the accumulation of superoxide. Accumulation of superoxide activate antioxidant system in plant, so the superoxide will convert to H2O2 by superoxide or metabolite. Then H2O2 will neutralize to water by catalase or APX. H2O2 is the most stable ROS. It can also cross cellular membrane. High concentration of H2O2 can cause oxidative stress and lead to the molecular or cellular damage and cell death. Therefore, plant develop a scavenging system to neutralize H2O2 by expressing specific peroxidase enzyme in mitochondria, chloroplast, microbody, and cytosol. ATAPX1 is a major peroxidase enzyme in cytosol, which is responsive to any abiotic stress. However, ATAPX2 is also cytosolic peroxidase, which is only responding to warmth, highlight, and heat. However, contribution of ATAPX2 in metal stress still unknown. Therefore, here we want to evaluate whether APX2 play a role in neutralizing H2O2 in nickel stress. For this purpose, we use APX2 mutant and overexpression line to test this hypothesis. We isolate two tDNA mutants from Arabidopsis Biological Resource, USA. The tDNA insertion was identified by PCR genotyping. PCR using specific gene primer identified signal in the wild type, but it was absent in two APX2 mutant. While PCR using specific gene and tDNA insertion identified the signal in the APX2-1 and APX2-2 mutant, indicating that the tDNA was inserted in the first exon of APX2-1 plant, and the tDNA was inserted in the fifth exon of the APX2-2 mutant plant. The ATAPX transcript RNA was identified in wild type APX2-1 and APX2-2 mutant using RT-PCR analysis. The signal was observed in wild type, but the RNA signal was absent in the APX-1 and -2 mutant, indicating that the APX-1 and APX-2 is knockout mutant. The mutant do not show any phenotype difference compared to wild type under normal or unstressed conditions. To generate overexpression line, we constructed APX2 cDNA under 35S promoter in P-GRIN2 factor. The construct was transformed to Arabidopsis thaliana using Agrobacterium tumefaciens method. In Western blot analysis result, the protein was detected in representative overexpression line 1 and line 2, but the signal of protein was absent in the wild type, indicating that the APX2 protein is constitutively expressed in transgenic line even under normal condition. The overexpression line do not show any phenotype compared to wild type in non-stress condition. Here we challenge wild type overexpression line and mutant to the heavy metal stress by growing the seedling plant in media containing nickel for 2 weeks. The APX2-1 and APX2-2 mutants show growth retardation and browning leaf compared to overexpression line and wild type, indicating that mutant line is more susceptible to nickel than wild type or overexpression. Overexpression line show more green leaf compared to wild type and mutant, indicating that overexpression line is more tolerant to nickel than wild type mutants. As conclusion, a high concentration of nickel in media might trigger the accumulation of H2O2 indicated by browning leaf in Arabidopsis. APX2 overexpression helped to eliminate the excessive amount of H2O2 and confer heavy metal tolerant to Arabidopsis. The absence of APX2 in mutant line reduced the capacity of seedling to neutralize excessive H2O2 in heavy metal stress. APX2 possibly play a role in scavenging H2O2 under heavy metal stress. Lastly, thank you for your attention.
Thank you, Paritno, for promising research and clear presentation. And then the last video presentation from Ms. Eva Agustriana. The screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for the chance. I'm Eva Agustriana and I would like to present our work entitled Optimized Expression of Large Fragment DNA Polymerase I from Geobacillus thermophilus in E. coli expression system. BST polymerase is a DNA polymerase obtained from Geobacillus thermophilus, which possesses polymerase activity as well as thermal stability, strand displacement activity, and high sensitivity. Such characteristics make this enzyme being employed in LAM amplification, which is one of the prominent methods for detection, including the detection of COVID-19. This is due to the ability of this method to be performed under isothermal conditions, neglecting the need of thermocycler, and the reaction is less time-consuming compared to RT-PCR. However, despite the potency of this enzyme, study about the production of this enzyme in Indonesia is still limited, let alone the optimization. For those reasons, this study is designed to perform optimization of expression condition of gene encoding BST polymerase. This study serves as a preliminary study for scale-up production of this enzyme. To achieve the goals, in this study, we performed several steps. Initially, synthetic gene encoding BST polymerase was transformed into expression host, and subsequently, positive transformant was cultured into LB broad media supplemented with kanamycin. And then, the culture was induced with IPTG once the optical density reached 0.8 to 1. After 16 hours of incubation, the cell pellet was harvested and being subjected to sonication. All fractions were then analyzed using 10% acrylamide gel. For optimization, once the positive transformant was cultured into LB kanamycin media, it was subjected to several variations. We first varied the IPTG concentration ranging from 0.1 to 1.4 millimolar. Subsequently, the incubation was conducted and the crude enzyme was subjected to analysis onto 10% acrylamide gel. Protein concentration for each treatment was estimated by imaging analysis. Once the optimum IPTG concentration was obtained, this was used in the next step of incubation time optimization. For this step, upon the addition of inducer, the culture was incubated for 6 to 24 hours of incubation. The optimum incubation time was determined by estimating the protein concentration for each treatment by imaging analysis. The optimum incubation time was applied in the next step of optical density optimization. For this purpose, the induction was performed at various optical density ranging from 0.4 to 1.1. And after the induction, the incubation was continued and the concentration of protein was estimated by imaging analysis to determine the optimum optical density. Upon obtaining the optimum condition in flash scale, we continued the optimization of agitation and aeration in 1 liter fermenter scale. The steps conducted were similar with those in the flash scale. However, the agitation and aeration were optimized by varying the agitation from 50 to 150 rpm, while the aeration was varied from 2 to 4 ppm. Upon obtaining the optimum conditions of agitation and aeration, the production in 1 liter fermenter was conducted and the crude enzyme obtained was purified using IMEC by using his trap column. The purified enzyme was subjected to activity assay. And here's the result of our study. We first conducted the SDS page analysis of expression of BST polymerase under initial conditions, and as can be observed from the SDS page analysis, the enzyme was successfully expressed as indicated by the black triangle, corresponds to to a protein band of around 67 kilodalton. However, the soluble fraction still required optimization, and for that reason, in the next step, we perform several optimization. The first optimization we conducted was the inducer concentration, and as can be observed in the figure, the increase of IPTG concentration of up to 1 millimolar was followed by the increase of protein yield. Further increase, however, impacted on the reduction of the protein yield. For that reason, IPTG of 1 millimolar was applied as the optimum concentration. In the subsequent step, incubation time was optimized. And according to the data, 6-hour incubation after the induction process was sufficient to induce the highest protein yield. Prolonging the incubation time, however, did not increase the protein yield. For that reason, in the next step, we employed 6-hour incubation as the optimum condition. Moreover, the optical density at which the induction was performed was optimized, and the result showed that induction conducted at the optical density of 1.1 was the optimum, equal to 2.8 times than that when using initial conditions. Upon obtaining the optimum conditions in flash scale, we perform the optimization in a 1 liter fermenter scale. As can be observed in the figure, at all cases, in a 1 liter fermenter scale, it was found that the optimum yield of protein was obtained at 12 hours of incubation instead of 6 hours in the flash scale. This result was different from that obtained in a flash scale. However, the discrepancy could be understood since there was a big difference in volume. And for this reason, incubation in a 1 liter fermenter scale was performed for 12 hours. Moreover, agitation and aeration are also optimized because those two factors are significant for liquid turbulence. Improper level of agitation could result in poor oxygen transfer and thus inhibit the cell growth. In the study, agitation at 100 rpm and aeration at 3 ppm resulted in the highest protein yield. In the subsequent step, the production of BST polymerase was conducted by employing these optimum conditions. And here is the result of the production of BST polymerase in 1 liter fermenter. We performed the analysis of SDS page and also Western blot. And based on the figures, BST polymerase was successfully expressed and purified to provide protein of 11.7 milligram per liter culture. The yield of purification is 48% with over 8-fold purification. And the activity assay conducted showed that the purified enzyme had polymerase activity much higher than that of the crude enzyme. This finding indicated that this approach is potential to be applied in BST polymerase production. And as a conclusion, in the study, BST polymerase was successfully expressed, purified, and produced in 1 liter fermenter. The optimum conditions for expression were as follows. Induction using 1 millimolar IPTG. The induction was performed at the optical density of 1.1. Incubation was for 12 hours under the agitation of 100 rpm and aeration of 3 ppm. Approach used in this study is potential to be implemented in the production of BST polymerase. And that's all. Thank you very much for the attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ms. Eva, for valuable research and good presentation. Dear all participants, all presenters have already delivered their video presentation. In summarize, from 6 presenters, we have Pak Tulus and Pak Sutikno conducting research in Bali cattle, and then Ms. Nisa and Pak Rikno in transgenic plant, and then Pak Witten and Ms. Eva in recombinant enzyme. Okay, that summary will bring us for Q&A. So I think we still have plenty of time for discussion. So please raise your hand or write down your question in the chat box and to whom the question will be addressed. Okay, I have one question from Endang 3. address it to Pak Sutikno. Is your finding can be applied for other beef cattle? And what kind of meat quality you got positive result from your research? Please, Pak Sutikno, would you answer that?
Okay, I can answer that. Good, thank you, Prof. Endang, for your question. Firstly, this is gen is candidate marker for meat quality, especially for score marbling in Hanwoo and Wagyu cattle. But in case in our cattle in Bali, the SNP is monomorphic. But in other hand, we find new SNP which was monomorphic in other cattle, but in Bali cattle is polymorphic. I believe it can be a candidate marker in Bali cattle, especially in Bali cattle, maybe not in other cattle, because in other cattle is monomorphic for this SNP. Maybe that's my explanation. Thank you.
Thank you. Thank you, Pak Sutikno. Okay, thank you, Pak Sutikno and Bu Endang. And then the other question from Pak Win to Miss Eva. Did you still use LB medium for the optimization experiments, or already use a medium for high cell density culture of E. coli? Please, Miss Eva, would you like to answer that?
Thank you. Thank you very much to Ms. Fatima, and thank you to Mr. Win. Yes, we did use LB medium for the optimization expressions, not using the high cell density. Yeah, we still use the LB medium, Ms. Win.
Is it enough, Pak Win? Or do you still want to asking more? Okay, thank you. We have other question. Okay, from the Pak Dani Setiawan for Miss Eva. If you sell your enzyme produced from this scale, in terms of price, how does it compare with available commercial enzymes? For Miss Eva, please answer.
Thank you very much to Mr. Dani. Actually, we still do not think of commercialization, since in this research the components we use were commercial, and it is not possible for us to bring it to commercialization if we use the current conditions. If we are to commercialize the production, we should use the host expression system, the non-commercialized expression system. However, for the compared to the commercial enzymes, we actually haven't performed it yet. But if we could develop a non-commercial host expression system and using the optimized medium, which is not the synthetic medium like LB, we might be able to provide a more economical BSD polymerase compared to the commercial one.
Is it enough, Pak Dani?
Yeah, I'm just saying, if not that you have to. I mean, if you try to sell it right now, like, how does it compare in terms of price? Because like enzyme price in our country is overpriced, right? So I think even if it's expensive, I think it might still be competitive. I don't know. So it's just a hypothetical question, not a real intent.
Okay, thank you, Pak Dani and Miss Eva. Yeah. Do we still have any other question? Please raise your hand, or you can write in the chat box. Or maybe one question also for Bu Eva. Do you have any strategy for increasing the yield, since in the purification is resulted around half, 48%. Do you have any other strategy for that? Yes.
Yeah, we are currently trying to optimize the medium also for the production performed by our team. And actually, the 11.7 milligram per liter culture was relatively low, small quantities. However, if we compare it to other reported studies, there was study reported the production around 3 milligram per liter culture. So it's good enough, but we need to improve the yield, maybe by the optimization of the medium.
Okay, thank you. There's one raised hand from Pak Win Kusaryato. Please, for Pak Win, your question.
Yeah, I just want to make a comment about that. And actually, it is a long way to go if you want to produce protein from E. coli, because usually you still have or you still use plasmid. Yeah, and in the industry, usually you need to integrate the gene into the genome of E. coli for the stable expression of the protein, and then using an optimized method for the fermentation. And the way is using high cell density culture. Using flux, for example, you only reach around 2 gram. An OD, an OD of 2.0 maximum, but using high cell density culture, you will have around 20 in optical density. So you will have much more protein produced by high cell density culture, of course, using minimal media, not using full media like LB or supergrowth media. So usually, like my research, for example, there is still a long way to go in producing the recombinant protein using the OCLI systems. This is my comment to the question. Thank you.
Okay, thank you, Pak Win. And so for Wifa. Do you have other questions, please? Or maybe I can ask for Pak Rigno. From the result is a kind of promising research. However, as we know that Arabidopsis is still the mother plant. So for further research, do you have any plan to develop it in other plant? If yes, in what plant? And how about the IPR? Since the construct you use have been obtained from the Ohio State University. So perhaps you have any plan for develop your own construct in BRIN? Please, no.
Thank you. We obtained the only mutant from the Wisconsin, from the USA, but for all construct like gene and plasmid for agrobacterium, all we constructed here. Now, our target is not Arabidopsis. Most of us working in crop plant like for dicot, we work in soybean, and for monocot, we work with maize and sugarcane, like that. But maybe we will choose other gene not from Arabidopsis. Some gene related to the biotic stress we got from soybean now. So here, I think is it difficult to work with Arabidopsis? We need a specific chamber or something that is not available in our office. So no other choice. We have to work with crop plants like that. Thank you for question.
Okay, thank you. And actually, I am curious. I would like to know why in this research is focusing on nickel rather than in other heavy metals. Does it relate with the reclamation of the nickel mining in Indonesia, such as in Sulawesi, or about the Tesla, something like that? Is it related with that?
Why we are working with nickel? Because like cadmium or other metal which was well known, there is many publication about that. But for nickel, it's not many publication for nickel stress. So that's why we are working on that. But before I didn't think about mining nickel in Indonesia. It's not our not in our thinking before. We just thinking that publication in nickel tolerant is not so many. So because of that, we are choosing nickel, not cadmium or others metals.
Okay, thank you. Are there any questions? I think I have a question for Miss Nisa. Are you there? I don't see Miss Nisa here.
Yes, I'm here.
Okay. Actually, it's an interesting study for phenomic study. Hopefully, in the near future, that BRIN will have this kind of equipment in this research. The transgenic plant that under saline stress that we found here, it seems less affected compared to other plants, as you mentioned before in the introduction, that in Arabidopsis, cotton, and others, the FEP1 gene is increased in their shoot growth. And how do you explain about that?
Well, actually, first of all, thank you for the question. And this was also the question when I was discussing with my supervisor, because I said, well, this is interesting. We read from the literatures that there are enhancement in biomass and the growth of the plants when they are overexpressed with AVP1, but how come we get different results? And then, well, actually, we haven't got the answer yet, but probably it has something to do with the tolerance of the IR64. So probably it has moderate tolerance to salinity, the IR64. And in this case, that the overexpression doesn't give much of enhancements of the growth, because we think that initially the IR64 already have a tolerance, moderate tolerance to salinity. Probably that's our first guess, but we still have much to do in order to find the real answer to that.
Okay, thank you. Hopefully, yeah.
Yeah.
And then do you have any question for other people?
Sorry. Sorry, were you asking me?
Yeah. Do you have any other question for other presenter?
I don't have any in mind right now, so I think that's it.
Okay, hopefully successfully for your research. Okay. And then
thank you.
We can have other question for Pak Tulus Maulana. Are you there? Pak Tulus Maulana. Okay. Actually, I'm not familiar with these bulls of cattle and veterinary word. So it was just a simple question. What is the you mentioned here about the candidate and certified bulls? Actually, what is the difference between these? And then in this METO status team TMRE as a marker, are there any information of what kind of other markers that have been used so far before you mentioned it as a marker? Okay, thank you. Okay, do we have other question here, please? Or it's already lunchtime? Okay, probably one more question for Pa Sutikno. I would like to ask, how do you perform your analysis? I mean, the association analysis. Did you use any software, any program? So if yes, please mention here what program did you use? And since so many, actually association analysis program for SNP and traits is already developed and they are available for free, such as Tussle, R package, and others. So probably you can answer about that.
Okay, thank you for the question. We use SAS with procedure GLM, linear model. For this analysis, we do not use the same what is called the same sample. Maybe we find 3 genotype, but sample for each genotype is not same. With JLM can calculate, but for RAL or RAC is cannot calculate, but JLM can calculate. So we use JLM in SAS with SAS program.
Okay. Okay, thank you. Maybe you can put it in the thought before. Yes.
Thank you.
Okay. Do we have any other question? Okay, probably for Pak Win. Are you still there?
Yeah, I'm still here.
Okay. Actually, this research is quite interesting. You have already starting from the construction, expression, also purification, and then also the in silico analysis for protein modeling. Hopefully, in the near future, the cryo-EM is already here in BRIN. So the research will be more comprehensive. And of course, the enzyme activity should be conducted also in the experimental research. My question is only one is mentioned earlier in the slide that the amino acid similarity was having low identities around 60 to 80%. Would you like to explain more about that?
Well, it is also related to a pattern. Usually, if you want to have a pattern, there is a low similarity with other enzyme, for example, in amino acid sequence. So that's why I search for enzyme, in this case L-arabinose isomerase, which has low similarity or identity with other arabinose isomerase from other organism for pattern application later on. And the next step will be to enhance the enzyme activity. We were able to enhance the activity of L-arabinose isomerase from Cuiobacillus stereotomophilus by almost 3 times. So the result of that experiment will be used also in the enhancement of this enzyme activity of L-arabinose isomerase from Thermotheca termarum. And of course, as I said before, it's a long way. Yeah, to go. Usually, you need to insert the gene into the genome of E. coli or other expression hosts. For example, Bacillus licheniformis, for example, and then to perform high cell density culture and to obtain the enzyme in high amount to reduce the price of the enzyme, and then to apply the enzyme in the production of tagastose from the galactose. Yeah, there is already an example in Europe, in Belgium, a company called Nutrilab, and we already produce tagatose by enzymatic processes, and they sell the product in Benelux country and in France in different kind of products like chocolate or like beverage, similar to in Indonesia, but in this case they replace glucose with the tagatose.
Okay, I think it's almost our time. I think we don't have much time again. So thank you for all presenters and participants for your attention and participation. We apologize for any inconvenience. We will continue with the rest of the seminar agenda. So would you please kindly return to main room? Thank you. Wish you all the best and stay healthy and have a great day. Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Thank you.
wabarakatuh.
Thank you, Ms. Fatima.
Recording in progress.
Welcome back to the main room, ladies and gentlemen. Now, we would like to invite Ms. Novio Ayusril, Product Specialist, Protemix Nutrilab, to share the presentations. Ibu Novia, good afternoon. Thank you for joining with us. Can you hear my voice?
Hello.
Yes, time is yours.
Thank you, everyone. Let me give me one moment, please. Yes. Do I need to present in English or should I just in Bahasa?
It will be better in English, Ibu, because the participants not only coming from Indonesia. Thank you.
Okay. Thank you. Yeah. Good afternoon, everyone. My name is Novio. I'm a product Nutrilab Pratama is one of distributors bio life science products in Indonesia. So our office is in Kebayoran Lama, South Jakarta, and here you can see you not only can find us in Jakarta, but we also have office like branch office in Bandung, Semarang, Yogyakarta, Surabaya, and Bogor. So for BRIN especially, we have ourselves here, which will help you guys whatever you need from us. So you can see in the right side that we can support you guys with these products, but mainly for Nutrilab, you can reach to us for Gitco product, in vitrogen, and Thermofisher scientific products. This one especially for you guys who are doing biology or biomolecular research and also cell culture research. And also, not only that, we have several products which support your like general instruments such as centrifuge, cortex, etc. So we have also bio-based and also Hanil from Korea and Clever Scientific from UK. For the plastic consumables, we have products such as TPP, Tarfon, Nest, etc. So this is the short introduction from me for our company, and maybe I will move to the next slide about what is the product or what can we do or what can we help you with your research here. So we have several divisions in our company, mainly supporting this group of research. First one is molecular biology. You guys who use PCR reagent or like real-time PCR, and also our best product is qubit, come from in vitrogen products. And also for protein biology, such as antibody, ELISA, Western blot products, especially for this product line, you can reach out to me if you want to have more information about protein biology. And then we also support you guys with synthetic biology from Thermofisher scientific. So if you're doing expression protein and also cloning, you guys can reach out to us how to order, or you want to try like positive control for your real-time PCR, you can also reach out to us. And then cell biology, our main product, which is from Gitco. So we are the only one, like the only authorized distributor for Gitco product line in Indonesia. So if you are doing cell culture, you can also reach out to us. And then the last time is cell analysis product, which is microscope, fluorescent microscope, flow cytometry, and also high content staining. This one is the for protein analysis flow. As I mentioned before, that anyone who use protein like to do such analysis like Western blot, mass spectro, we have these many products that can support you guys. So from the extraction of the sample to the protein cleanup like dialysis, desalting, concentration, and also the downstream applications such as I mentioned is the stage Western blot workflow. We have here like Western workflow that maybe you guys also have heard before, that usually you takes 2 days to do Western blot, right? We have product line that can use that you guys can use only in 4 hours. You can get the result. So if you guys are interested, you can reach out to us for this application. And the last time is the analysis. Next is PCR workflow. As you can see here, we have we can support you the whole workflow from the extraction of DNA or RNA and then the quantitation. So here you guys may be familiar with nanodrop or spectro. So here we have a qubit fluorometer which use fluorometer detection, and also we have the master mix for both conventional and real-time PCR, and then of course the PCR instruments, and also the electrophoresis system here which use power snap. Next, as I mentioned, that we support you guys with the GIPO cell culture media. Maybe you are familiar with our GIPO product already. So you can see that from the isolation and then to the storage media, you can see here that we can support you guys with this. This one is our like high-end product, which is high content screening. Maybe you are familiar with this term, right? That you can use this microscope, fluorescent microscope light plus microplate reader, and also that analysis data for your drug discovery. So you can see that this, for example, you have like a chemical compound that targeted RNA or other target in your cells, and then you want to try to check whether this drug is actually works for your cancer or for other diseases. So you can try with our high content screening CX7 LBL Pro, and then maybe for the next level you can also image the this work with bioluminescence. This is, I think, my last two slides for our general equipment, such as I mentioned before, right? The freezer, centrifuge, like UV cabinets, and also all the stuff that you are familiar with in your lab. You can also, if you want to purchase your unit, you can reach out to us, or maybe some consumable or like accessories that you need, you also can reach out to us. I think this is my last presentation. So this one not only for bioscience products, we in Nutrilab also support you guys with bioproduction products. Maybe next time, now you are doing like the R&D step, right? And maybe in the future you want to produce your own like vaccine or your own drugs or protein recombinant. We can support you guys with these product lines from research development, bioreactor or fermenter. Also, we have this for biofactoring. Maybe you guys doing cell culture, which like adherent cells. You can also use our biofactory from next. And the last time is for purification of your protein in large scale. Usually you just only use like spin column or ASTA, right? You're familiar with those stuff. Here we have the resin, which can help you guys to use it in your purification system. Maybe that's all from me. This is I'm very grateful for this opportunity to present to you guys all our products in Nutrilab Pratama. So thank you so much. Yeah, see you guys.
Thank you very much, Ms. Novio, for comprehensive explanations. As what mentions, ladies and gentlemen, you can contact NutriLab directly for further informations of their services and also their products. So, distinguished guests, ladies and gentlemen, as the time is now 12:22, we will have a break for approximately an hour and 8 minutes, I guess. And please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Once again, we will have a break for approximately an hour and 7 minutes from now on. So please kindly back to the Zoom meeting at 1:30 p.m. Jakarta time. Thank you and have a nice lunch.
Halo Arnal. Hello, Uni.
How are you?
Hey, fine, fine. How are you?
Very
Good to
good.
see you.
Yeah.
Great, great.
Yeah. So what's your? Are you already okay now?
Yes, I'm fine. Yes.
Okay, okay. So how long?
Shall I just
Yeah. Start your
share my screen?
Yes, yes, please.
Because I'm trying to do that with two computers, so I have to see how this is working. Just a second.
Yeah.
Stop sharing, because
Enggak, share screen. Are you familiar with Zoom, Arnold?
I'm sorry?
Are you familiar with Zoom?
No, we are normally using mostly Teams.
Teams. Oh, okay.
But I try to share my screen.
Yeah.
But I cannot. But I get the message. You cannot start screen sharing while the other participant is sharing.
Okay. So the organizer will stop this. Okay. Now it's already okay. You can
now it
try.
should be possible.
Yeah.
Okay. Let me just try. Yes.
Yeah, I already see your presentation.
Yes, but there's still some annoying thing in here.
No, it's.
Let me check. There it is.
Yes.
Yeah, it's working. Perfect.
Okay.
Good. Now, I'm glad it's working, Uni. So this is the way we will do it.
Yeah, Arnold. So after the break, around 6 minutes before your talk, there will be a parallel session, and you can choose which room do you want to.
Yeah, I will check.
Yeah.
I want to go.
Yeah.
But just a second, I'm trying to get out of the screen sharing now.
Oh, gimana caranya kamu?
Because that's not working yet.
Ya udah. Gimana caranya kalau mau keluar dari share screen?
Here.
Yeah. Share. So in this
I'm still sharing, eh? Or not?
No. You already stopped sharing your screen.
Okay, then it's fine.
Yeah.
All right. I guess it will work.
Okay, thank you very much, Arnold, for your participation.
No, I'm very happy to do that. Thank you for the invitation. It's nice.
Yeah. In this afternoon, there will be only you presenting in the plenary. Yeah, plenary speakers.
Yeah.
So you will have a lot of time for presentation and the discussion. Yeah.
Okay, fine. Yes. Great.
Okay, I'll not see you later.
See you later then. Okay.
Yeah.
Bye.
Hello. Okay, hear my voice loud and clear.
It's perfect, Mr. Andri.
Okay, thank you very much. Let's see. Dr. Maulida is already here. Dr. Gulilis is already here. Wait. Dr. Ira, not here yet. And Titis is already here. Nah, okay. Please wait a moment. Okay, right now we are waiting for Dr. Ira. So maybe she can join later, I think, because it's already 1:30. So we can start I think we can start. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. So my name is Andri, and now allow me to lead this session, this parallel session as a moderator. So let's explain how the rule of this session. For this session, we are going to have 4 speakers on my list, and each speaker will present their presentation for about 10 minutes, and after that, we will have a Q&A after they are presenting their materials. We will have a Q&A session for about 20 minutes. So let us move to the first speaker. So the first speaker is Dr. Maulida. She will present her very interesting work entitled "What Indonesian Gene Bank: a framework for repositories and discovery for nucleotide sequence in Indonesia." So, Dr. Maulida, time is yours.
Hello everyone, my name is Maulida Mazaya. On behalf of our team, I would like to present our paper entitled "Toward Indonesian Gene Bank, a framework for repository and discovery for nucleotide sequences in Indonesia." We are from Research Center for Computing, National Research and Innovation Agency, or BRIN. Okay, let's start with the introductions. As far as we know, Indonesia is a mega biodiversity and becomes the hotspot in the world. Unfortunately, our richness of the biodiversity has not been well utilized. And most of our researchers, especially, they send their results of the biodiversity and other things, especially in the nucleotide sequences, to the World Nucleotide Archive dataset or databank. For example, NCBI from the United States or European Nucleotide Archive, or DNA Databank of Japan, where they make the ENSDZ, this is kind of the international standard for nucleotide sequences in the world. And recently, the National Genomic Data Center from China, they also follow them to build genome sequence archive. And this is actually our chance to build our own repository archive. But why we cannot do that before? The reason is only one, because there is no integrated public or private repository data bank, or we can call it a biobanks, to do analysis and to store our data sets for especially biodiversity and health in Indonesia, where the recently that our Minister of the Health started the initiative to build the BGSI for the precision medicine. But of course, this is not enough, because even BGSI 2, it still needs the repository data sets. It still needs the repository platform to store the nucleotide archive. Then so that they can run or they can do the precision medicine analysis for the further. Okay. So that's why in these works we propose the developments of the Indonesian Gene Bank, or we call it Indonesian National Nucleotide Archive, or INA. This is kind of the integrated knowledge base to provide the storage, computation nodes, and data server for data sequencing and analysis in the field of the life science, foods, and health. And this will be utilized using the computational infrastructure in the BRIN, that is the high performance computing. And we do hope that our platform, our framework, this can be globally accepted as a computing resource and users. So this is our proposed methods. This is the architecture theme of Indonesian Gene Bank, or INA. Here we have two main functions as a repository platform and the discovery platform. And we do have the kind of tools that support these frameworks. And here, users can submit their data set, including the sample extraction or sequencing RNA DNA, following the online system analysis for sequencing lab process, this one. And of course, after users submit their data, they cannot just do that. I mean, they cannot just do the more analysis after that, because they should wait for the data and knowledge validator. So we will validate their submitted data sets. If we will check that if their data sets already follow the international metadata ontology or not. And after that, if there is no problem, the data is valid, then the users can access their submitted data sets, the sequence data, and knowledge retrievals. And in the discovery platform, the users can do advanced analysis for doing like a basic pipeline, and to do more analysis in biodiversity pipeline or genetic resource utilizations, and in the precision medicine. But here, the precision medicine, we are collaborating with the BGSI. So this will be interesting in the future. And going to the result sections here, in the development of this repository, of course, we follow the international metadata standard. So we have architecture like this in our frameworks. We have the bioprojects, biosamples, bioexperiments, run, and archive. Bioproject here is defined as a collection of molecular projects for large-scale genome sequencing, assembly, transcriptomics, metagenomics, and others. And inside by project, we have some of biosamples that define as the collections of general informations related to the biological source, like the raw data and other things, where the biosamples has the many childs of the bioexperiments. And these experiments will be wrapped into one of bioarchive. And the next is knowledge-based implementations for data sequencing analysis. This is the process in the validations data or in the data creations. As we know that users submit 3 types of data set, right? And in the submission, in our system, we will integrate every submitted data here and then transform them to the metadata standard by employing uniroles roles. These uniroles we modify or we adapted from the uniprot. And after this system, the process finish, by the logical inference from the rule engine, then the results will be annotations based on these rules. So this annotation is we can say that is a validated data for the submitted data. So this validated data that will be saved or stored in our repository platforms. And this is our ongoing framework development. We can check it here in the URL. For the interface, we follow the international standard too. We have by project, by samples, by experiments, and by run here. And also the statistic of the data collections, where in this works, we employ MySQL standard and CodeIgniter version 4 to build this repository platform. Okay. And for the conclusions, in this study, we proposed the Indonesian Gene Bank, or we call it Indonesian National Nucleotide Archive, or INA. This is a framework for repository and discovery for nucleotide sequences in Indonesia to support the research and innovations in the life science, foods, and health also medicine. But of course, our works has kind of still improvement. We need to improve our works to consider the data safety, data sharing, standard repository, and others, following the international standard metadata ontology. And the second thing that we should consider is that the human metadata and ontology model and ethical code standard and their roles in Indonesia. I think that's all for the presentations, and thank you.
Okay, thank you very much, Ms. Mawida, for a very nice presentation. So we will going to move to the next presentation. So the next presenter is Dr. Ira Handayani. She will present her outstanding work entitled Mining Indonesia Microbial Biodiversity for Novel Natural Compound by a Combined Genome Mining and a Molecular Networking Approach. So, Dr. Ira, time is yours.
Good morning, everyone. I'm Ira Handayani from Research Center for Applied Microbiology BRIN. Today, I would like to present our published article with the title Mining Indonesian Microbial Biodiversity for Novel Natural Compound by a Combined Genome Mining and Molecular Networking Approach. Recently, the number of antibiotic-resistant bacteria have increased. That's why new antibiotic needed to overcome the antibiotic-resistant bacteria. Actinomycetes have been proven as an excellent source of antibiotic, as 2/3 of all known clinically useful antibiotics are produced by these filamentous bacteria. Sampling a unique biotope turned out to be an efficient way to discover novel antibiotic producers. As Indonesia is the third most species-rich country in the world, thus Indonesian soil should be a source for unknown antibacterial strains that might produce novel antibiotics. Therefore, in the study, we focus on bioprospective of Indonesian habitat for novel natural compound producer. The aim of the work is to investigate the potential of Indonesian actinomycetes to produce novel natural compounds. Sampling actinomycetes from conventional environments such as soil often lead to the rediscovery of known producers producing already known antibiotics. Thus, prospecting on novel habitat will be an efficient way to isolate novel antibiotic producers. In this study, soil samples from terrestrial habitat and marine habitat were collected from 3 different locations. Engeno Island was chosen as a sample site for terrestrial habitat samples, as it is a pristine island with a rich biodiversity and many endemic species, whereas Bali and Lombok were chosen as sampling sites for marine habitats. From this isolation effort, around 422 actinomycetes isolated from Indonesian soil samples. The study is divided into 4 parts. First is characterization of Indonesian strains. Next, genome sequence-based analysis. Third, metabolomic mining for compound identification. And the last one, linking genomic and metabolomic data. This is the workflow for the characterization of Indonesian strain. Actinomycetes from soil sample were isolated and then grown on medium suitable for their growth. Phylogenetic analysis and bioassay were then conducted in order to prioritization of strain for further investigation with genome and metabolomic mining analysis. From the 422 isolate, 16 samples saw the best antimicrobial activity. The 16S RNA phylogenetic analysis revealed that all 16 strains belong to the genus of Streptomyces. From this analysis, we also found that DSA67 and DSA51 is closely related to Streptomyces parvulus, a known producer of actinomycin D and most likely represent the same species. To find out whether the 2 strains produce actinomycin D, SPLC, and SPLC-MS analysis were conducted. SPLC and SPLC-MS analysis were applied to the extract sample of DSA67 and DSA51. The SPLC and SPLC-MS analysis confirmed that both of the strains produce actinomycin D. Therefore, both of strains were not included from next experiment with genome sequencing. And from bioactivity profile, 6 strains saw in a red box prioritized for genome sequencing. SAP27, base A79, base A7F, I3, I4, I5, and I6 were selected since they display antimicrobial activity against gram-positive and gram-negative bacteria. DSE77 and DSE71 were chosen because they saw bioactivity against at least 2 different gram-positive test strains. Next, all 9 strains were investigated further for genome and metabolomic mining analysis. The following is the workflow of analysis using a genome mining approach. First, the genomic DNA of the isolate was isolated and sequenced with Biotechnology. The result in the form of a sequence of genome data were then analyzed with type-strand genome server for identification of phylotype strands. The genome sequence was also analyzed with an anti-smash to identify the biosynthetic gene cluster. Then analyzed with BigScape to identify gene cluster family. The following are the results of the analysis with the type-strand genome server. Phylogenomic analysis with the type-strand genome server revealed that all 9 strands belong to novel Streptomyces species. This phylogenomic analysis also revealed that the strands isolated from the same habitat tend to belong to the same phylogenetic clade. From the anti-smash and BigScape analysis, at unique gene cluster family with a similarity of less than 60% to non-biosynthetic gene cluster identified in the gene cluster networking. 2 unique biosynthetic gene clusters were distributed among the strands SAP27 and DSA17 that isolated from organo soil sample, while 5 unique biosynthetic clusters were distributed among the strands I3, I4, I5, and BSA7F isolated from mangrove. This data indicate that phylogenetically related strands harbor similar unique clusters. Next, metabolomic mining was conducted for compound identification. For this purpose, the 9 Indonesian strands were each grown in 12 different soil and liquid medium, and then to be extracted and purified. The sample fraction were then analyzed with SPLC, Harusi, MS. The MS raw data were then analyzed with GNPS for compound identification. The last stage of genome mining is to link genome mining data with metabolomic mining to confirm whether the natural product was produced under laboratory conditions. From this study, the known and unknown compounds were detected on the extract sample. From the data, we found that the compound of napirodenomycin A produced by a strand that isolated from mangroves. Meanwhile, amicetin was produced by strand isolated from soil habitat. From this observation, it can be concluded that phylogenetically related strands produce similar compounds, and the novel Indonesian strands have the potential to produce novel compounds. From our study, it can be concluded that guided effort of bioactivity and taxonomic classification can lead to prioritization of the strand, and phylogenetically related strands tend to share similar biosynthetic gene cluster. Next, environmental conditions influence the type of species and the ability of the strand to produce certain compound. And the last one, genome mining and metabolomic mining can be beneficial to pave the way for more efficient discovery of new natural products. This is the end of the presentation, and thank you for your attention.
Okay, thank you very much, Dr. Ina, for very nice presentation. So we move to our third speakers. We have Dr. Puspita Lisdianti. She will present her very recent work entitled Chemotaxonomic Study of 2 actinomycetes, strain ID 03700 and ID 03721. So, Dr. Puspita Lisdianti, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, Today, I would like to share with you our research result on chemotaxonomical study of two actinomycet strains, ID 03700 and ID 03721. Actinomycet, that also called as actinobacteria, are prokaryotes belonging to the gram-positive bacteria saprophyte, usually produce large numbers of spores and form stable mycelium. Actinomycets widely distributed in nature, soil, and litter. Terrestrial and marine play important role in the decomposing natural organic compounds. In addition, actinomycets can produce numerous secondary metabolites such as antibiotics, enzyme inhibitors, and other bioactive compounds. About 2/3 of the antibiotics found so far are derived from secondary metabolites produced by actinomycets. Considering these advantages, some studies were conducted to obtain taxonomically new species of actinomycets, which were also expected to produce new bioactive compounds. In bacterial taxonomy, polyphasic approach that combine and evaluate different types of data obtained from microorganisms is very important to determine their taxa. Polyphasic approach which assesses the morphology, physiology, chemotaxonomy, and phylogenetic data is must during characterization and identification of microorganisms. Phenotypic characterization is important to understand the appearance, growth, and habitat of microorganisms. Chemotaxonomy study is essential for determining the most important differences between families, genera, and species comparatively. Phylogenetic data have revealed a hierarchy arrangement among the genera. However, this information cannot provide reliable data on the level of species or strains. It is no doubt in recent years, significant approaches has been made in systematics with the help of molecular biology studies. The data of ecology and genetics is also important to understand the behavior of microorganisms. All the characteristics has advantages and disadvantages. Therefore, combination of all studies is needed for valid identification. Cell wall composition of actinomycets is different from other bacteria. The cell wall of actinomycets is very complex. Therefore, analysis of cell wall component are the essential tools for actinomycet identification. During our study on isolation and identification of actinomycets from Ekakarya Botanical Garden, Bali, from 20 leaf litter sample by using rehydration and centrifugation isolation method, the total number of 85 actinomycets were found. Usually, actinomycets are abundant in soil samples, but we found also from leaf litter samples. The most dominated genus in leaf litter was the genus Actinoplanes, followed by the genus Catenuloplanes, Cryptosporangium, Kinospora, Lapilicoccus, Micromonospora, Pseudonocardia, Saccharopolyspora, and Streptomyces. From the blast search result, Two isolates of the genus Actinoplanes, ID 03700 and ID 03721, showed the blast value below 96% that indicated as new taxa. Therefore, we have interest to those isolates for further study. Methodology used for chemotaxonomic study are shown in this slide. First, we have to prepare cell biomass. About 500 milliliter of cell cultures of actinomycetes were lyophilized to get enough dried cell for chemotaxonomy determination. We use the dried cell for determination of diaminopimelic acid, cell wall sugar composition, isoprenoid kinones, phospholipid, and fatty acid composition in cell wall. The result of morphological characteristics is shown in this slide. Both isolates are gram-positive, aerobic, and have branches hypha. As shown in this figure, 14 days old isolate grown on HV agar medium were observed under a light microscope, and it was found that colonies grew in cluster and no aerial mycelium. Both strains have an orange colony. This figure shows the presence of single spore that appears to be aggregated on the mycelium substrate. Phenotypic characterization of both strains are presented in Table 2. Both strains grew well on 15, 20, and 25 degrees Celsius, and no growth on 10, 37, and 45 degrees Celsius. Both strains grew well on ISP2, ISP3, ISP4, ISP5, ISP7, and beneath agar. Both strains grew weakly in ISP6 agar. Both strains grow moderately in nutrient agar and water agar. No aerial mycelium and dissolved pigment were found in the two strains in any growth media observed. The purpose of the isolate to grow in different temperature and different media is for exploring their ability to produce diffusible pigment. The same isolates of actinomycetes were grown on different media can give different color. This indicates that actinomycetes have the ability to produce different pigment when culture media are changed. From the result of chemotaxonomy determination, the cell wall of two strains contain mesodiaminopimelic acid, same with other cell wall type in the Actinoplanes durhamensis. Both strains contain glucose as the main sugar content in intact cell. ID 03700 strain, in addition to glucose, also contain galactose and ribose. The cell wall of actinomycetes contains several types of sugar such as galactose, glucose, mannose, arabinose, xylose, and ribose, in addition to N-acetylglucosamide and N-acetylmuramic acid from peptidoglycan. Sugar composition play an important role in the identification of actinomycetes sporulation that have mesodiaminopimelic acid in their cell wall. The main menakinone of isolates is MK9. The phospholipid content of the cell wall indicated that phosphatidylethanolamin was detected in the cell wall of both isolates, while ninhydrin positive phosphoglycolipid only detected in strain ID 03721. This indicates that the two isolates belong to the P2 phospholipid group. Phylogenetic analysis based on 16S ribosomal RNA gene sequences reveal that both threads located in the cluster of the genus Actinoplanes. The genus Actinoplanes is a member of family Micromonosporaceae. Based on 16S ribosomal RNA gene sequencing comparison, ID 03700 and ID 03721 was closely related to Actinoplanes durhamensis type strain and Actinoplanes ferrugineus type strain with a similarity value around 97%. Actinomycetes, Actinoplanes cibodacensis and Actinoplanes tropicalis were actinomycetes that isolated from Indonesia. The genus of actinomycetes is rare actinomycetes, belongs to the family Micromonosporaceae. Some strains produce phalienamine precursor to the anti-diabetic drug acarbose. Some strains also produce the antibiotic validamicin, teicoplanin, and ramoplanin. Currently, the genus Actinoplanes comprises of 52 recognized species. In conclusion, the result of morphological, phenotypic, and chemotaxonomic observation, actinomycete strain belong to the genus Actinoplanes from the family Micromonosporaceae. Chemotaxonomy play an important role in regulating intertaxa connection, especially in actinomycetes biosystematics. Chemotaxonomy examines biochemical markers such as amino acids and peptides, lipids, polysaccharides, and related polymers, and other complex polymers compound to find the distribution of members of different taxa, and all this information is used for classification and identification. Chemotaxonomical characterization of actinomycetes should be conducted as a routine work to describe new taxa in this bacteria. Thank you very much for your attention.
Thank you very much, Dr. Puspita Disdianti. So very interesting work and very nice presentation. So for our last presenter, we have Ibu Yalni, and she will present her recent work entitled In vitro germination and micropropagation to support ex situ conservation of Calamus draco in Aceh. So Ibu Yalni, time is yours.
And micropropagation to support ex situ conservation of Calamus draco in Aceh. Introduction. Rattan is a non-tree forest product with high economic value. 600 species of rattan from 13 genera in the tropical rainforest of Southeast Asia. Calamus genus has 115 species and 8 species produce resin. Calamustraco grew in Sumatra and Kalimantan, produce blood red or dark red or jenang or dragon's blood. Jenang used in industries as ceramics, marble, stones, tools, wood, paper and pharmaceutical industries, antibacterial, antifungal, and antioxidants. Calamustraco was listed in IUCN Red List of Threatened Species. Doesn't include as a protected plant species. In Aceh, jenang population is decreasing due to deforestation and inappropriate harvesting, so need for serious attention. Propagation. Natural propagation of Calamustraco conducted by generative use seed, but in field observation, difficult to get fruit. The species seriously at risk of extinction, therefore need to add other alternative to overcome these problems. Plant tissue culture is an important technique alternative to produce seedling in large mass in a short time. An effective way to plant propagation, disease elimination, plant improvement, production of secondary metabolites, and in vitro conservation of plant, endangered, threatened, and rare species. Objective of this study to evaluate the effect of sterilization and additional plant growth regulator of benzyladenine in the propagation medium treatment on in vitro seed germination and propagation of Calamustraco. Plant materials. Calamustraco's seed originally from PD district, Aceh province, used as plants. Location, the tissue culture laboratory, CPTI, Yogyakarta, Indonesia. Sterilization, use alcohol 70% for 5, 10, 20, and 30 minutes, following the soaking in sterile water for 30, 60, 120, and 180 minutes. For seed germination, use MS0. For shoot multiplication, use MS medium plus BA 1 to 4 milligram per liter. Acclimatization. Data analysis. Analysis of the percentage of sterile culture calculated using the formula. Percentage of sterile explants is number of sterile explants divided number of culture explants times 100%. Analysis of the percentage of germinating seeds was calculated using the formula. Percentage of germinated explants is number of germinated explants divided number of explants times 100%. Result and discussion. Sterilization. The percentage of sterile explants obtained is 100%. Seed germination. The percentage of seed germination obtained is 70 to 95%. The best treatment for seed germination was sterilization with alcohol 70% for 30 minutes and soaking by sterile water for 120 minutes is 95%. The result study are highest than other studies as conventional germination of Calamustraco 80%, Nocoratan 91.98%, karbohidrat 88.67%. Seed germination. Table 1 showed the effect of the different sterilization treatments on the germination of Calamus draco seeds. Figure 1 showed in vitro germination of Calamus draco. Fluid of Calamus draco without skin germinated seeds at 3 weeks old and sprout culture at 6 weeks old. Shoot multiplication. Plant growth regulator PA 1 to 4 milligram per liter was not be able to stimulate shoot induction. Presumably, the tissue used as a plant is part of the whole plant. Growth regulator treatment are still used for growth apical dominance, not for the formation of new shoot. Apical dominance, the apical meristem is faster and more dominant than the lateral meristem, so that it inhibits the growth of the lateral shoot underneath. Cutting the apical meristem is the solution to break the apical meristem and let lateral shoot grow normally. The addition of plant growth regulator seems can increase weight of the plant, not for propagation. Acclimatization. In acclimatization media using a mixture of sand, ratio soil with a ratio of 1:1, the seedling had several leaves and was able to grow perfectly in age of 12 weeks observation. The germinated seed used for acclimatization showed good growth on germination media. The results showed that in 12 weeks observation, 90% of the acclimatization plantlet were successfully survived after 16 weeks of observation. 10% plantlet was found died in the early acclimatization stage because the presence of fungi. Conclusion. In the study was obtained seed sterile explant reached 100% from all treatment sterilization. The highest percentage of seed germination is 95%, obtained from sterilized using 70% alcohol for 30 minutes following the soaking in sterile water for 120 minutes. The treatment of BA 1 to 4 milligram per liter was not be able to shoot multiplication until 16 weeks. The highest percentage of plantlet acclimatization obtained is 70% on 16 weeks old. That's all. Thank you.
Thank you very much, Ibu Yelni, for the nice presentation. So we are going to move to the question and answer session. So please raise up your hand or click the raise hand icon if you want to convey your question to the presenter directly, and or you can type your question in the chat box. So and please state your name and institution and to whom your question directed to. So is there any question for the presenter? Also, there's one question from Dr. Ira Handayani to Dr. Maurida. So, the question is to be able to publish our research related to gene or genome sequence data. We usually have to store it in the public database, such as gene bank platforms, such as NCBI. Can we also store the data and publish it in the database? Dr. Maurida?
Okay, I will answer this. Yes, of course, you can, because the target of our work is in the future that especially for Indonesian researchers can submit their data set or the results of the research results in a database. But of course, we still need more time to make our platform better. Yeah. And the target is to develop everything will be finished within the next year, inshallah, like that. Okay, thank you.
Okay, the answer is yes, Dr. Ira. So, Dr. Ira, do you have any other comments for Dr. Maulida?
Okay, thank you. I have another comment. I just wonder when it started. I mean, when your team started to keep the genome sequence data?
We starting from this year, we are developing the platform, the information system, and others. So actually, in the end of this year, I heard from Parifki, my supervisor, that the genome sequencing can be started.
Oh, okay. Yeah.
Thank you.
Okay, thank you for the answer.
Sorry. Is there any other question for the presenter? Not yet. I think so. I have a question for Dr. Maurida. So this is a very interesting work actually. So just yesterday we had a discussion with our colleagues from Japan, from JICA, and we have we are going to have research work, and then they asked, because our research later they will produce like a gene sequence. So they asked us about how Indonesia has like a platform for storing the gene sequence, and at that time we cannot answer that clearly, because I only know we have the RIN, only RIN repository ilmiah nasional, maybe you know that. So we only have a RIN for storing the data. So at the time I cannot answer the question, and after hear your presentation, maybe I can show them that we have a platform to store the gene sequence. So the question is about the data ownership, because the Japanese side they concerned about the ownership. They want the data can be published to public. So for that kind of the ownership, I mean, who will own the data if we store the gene sequence to the INA platform? Can you make a comment on that? And also, what's the difference between INA and also RIN? Because in RIN, I think we have a similar storage system or something. Maybe can you make a comment on that?
Yeah. Okay. Very nice questions. So I will answer one by one. Related to the data ownership, since we will follow the international standard metadata, of course it will be open, but it's not all data will be open just like that. Like you submit something, then everybody can access your data. It's not like that, because there is some rules that we should follow, and we call it. If you hear about fair sharing, it means that to submit the sequencing data set internationally, following the international standard, we should make it really open, and then data to be fair. That means can be accessed by anyone who need it in the terms something. So not just you can get it, but following the rules and accessible for everyone. And then anyway, it can be shared. And for the ownership, there is some rules and role. So don't worry about that, because we will follow the metadata standard. So that's the answer for the first questions. And the second is the difference between INA and RIN. Of course, it's totally different, Mr. Andre, because the structure and the metadata itself is different. In INA, we will follow like biosample, bioproject, and others. Like if you use NCBI or DDBC or European Nucleotide Archive, it will be like that. The structure of the interface, we will follow that. So automatically the metadata and the format will be different. Thank you.
Okay, thank you for the nice answer. Thank you very much, Mr. Rikno Harmoko. Mr. Rikno Harmoko, please.
Okay, thank you. I have a question for Dr. Maulida. I think this interesting topic that we have like depository program like this. But we know today there is many repository database like DDBG, EMBL, EBI, then NCBI or Phytosome or something like that, that they prove very integrative system from sequence, protein, and structure like that. So what is the difference between the INA system compared to those? Then what make this, how to say, your INA system will be at least better than that, like that? Maybe you can give answer, then how to say, we get more interest to that your systems. Okay, thank you.
Thank you for the questions. I will answer the second questions. At least better than NCBI. I cannot guarantee that because we are still under development, yeah, of course. But we do hope that we can be at least similar, or yeah, of course not exactly same, because NCBI and DDBC, they have built their system long time ago, and we just start this year. And the differences between INA and them is that most of our focus are for the Indonesian researchers, that we know that most of us submit our dataset there. And during this development, we have some discussions with the Ministry of Health, and they also think about especially the human genomics safety, data security, because Indonesia, if we submit our dataset to them, we do not know what will they do with our data. So this is the most important things for the data security. That's why it's better to have our own repository. So that's one reason. And the second thing is that not all general repository like NCBI and others has complete data set for our biodiversity yet. That's the second reason. And if we can manage our biodiversity and we can analyze and submit in our own repository, then in the advance we can analyze and do some more analysis and research in our own biodiversity, because our biodiversity is very rich and it's pretty yeah, what can I say, it's too much if we send our dataset to them than if we have our own. Yeah, thank you.
Okay, thank you.
Okay. Thank you very much for the question. Is there any other question? Maybe from the presenter to presenter, like, okay. Is there any other question? Okay. Not yet, I think.
May I have another question?
Okay, Dr. Ira,
Thank
please go
you.
ahead.
I just wonder. So the Inovasi is just for Indonesian or is
No. It's open for anyone, but we are focusing for Indonesian researchers at this time.
Okay.
But we will follow the international standards, so it will be open for public.
Okay, thank you. Thank you.
You're welcome. Thank you.
Okay, thank you very much. Is there any other? Okay, I wanna ask Dr. Ira then. Okay. Okay, Dr. Ira. So I really interested in actually in like your work. I mean, you use like bioinformatics tools and also use the like Pigscape and other tools that are actually that's free of charge, right? So there is a open that tools are open. So I want to ask you about or your opinion about it. Is it for me, like I haven't done the research of like kind of like yours, right? Like Bioinformatics and so on. So is it the learning curve, the learning curve it is steep, or I mean, is it really hard to do, or like it is easy because you have these tools, right? You have this tool in the internet, you can just use it, right? So can you give me your opinion about that?
Okay, thank you for your question. I think for me it's easy because before my master degree is bioinformatics, so I don't know if another person also same with me, but I think because they're all what is the book what is the procedure book or something like that if every platform or every bioinformatic tools, I think you can read it. I mean, manual instruction in there, so you can learn by doing. Because the informatic study usually used by learning by doing, not really teaching. Yeah, not really teaching, but if you use every day, I think you can make it better to use it. I think so. It's easy. It's just like words. If you learn, if you use Word platform or Word file every day, so you can use it more properly. I think something like this. Yes.
Okay. Okay. The point is that you have to learn by doing it.
It's learn by doing.
Okay.
Yeah.
Okay. Thank you very much for the insight, Dr. Ira Handayani.
Yeah.
So I have a question for Dr. Puspita Disdianti. So Dr. Willis, the term of like chemotaxonomy is like new for me, right? Because I just heard it from you. So can you give us an insight how this chemotaxonomy, I mean, what kind of data you can get from the chemotaxonomy, and maybe you can give examples. For example, I mean, I just don't get it how the chemotaxonomy work. Maybe Dr. Puspita, can you give us like more insight about chemotaxonomy?
Okay, thank you very much, Pak Andri. Yes, currently chemotaxonomy also be used for the characterization of plant and animals. Yeah, I think during the study of systematics of organism, sometimes we found some difficulties to differentiate between the species or strains. at that time, chemotaxonomy is really important to differentiate interconnectivity between strain, species, and then genera. Yeah, that's why. Yeah, chemotaxonomy. It's really we can know the deep insight of the microorganism, not only the appearance but also inside of the organism. Yeah, actually, in the microorganism, in the actinobacteria, it's really very difficult to differentiate each other. Yeah, we have to know the cell wall type and then also the protein. Yeah, it's like uh, to know the protein content, the cell wall type, and then also maybe the metabolite that they produce it. Yeah.
So maybe a certain species of bacteria or for this case actinomycetes.
Yes.
They can produce like specific compound.
Yes, right.
Okay. I see.
Yeah. And then we only know them by chemotaxonomy. Yeah.
Okay, I see. Okay, thank you, Dr.
You are welcome, Pak Andre.
Okay, maybe the another question for the Ibu Yalni. So, I think we all know that the plant that your plant is the rattan is in the IUCN Red List, and it is important. It is important to conserve those plants because it is endangered species, right? So we need a method to conserve them. One of the method is through the tisserculture, like your work, right? So I see that your work is not completely done yet because in your work you have I think there is no micropropagation, right? Because it fails to do the shoot proliferation. So maybe Bu Yalni, can you give me insight? Maybe what strategy can you give us a strategy so can you to succeed in my micropropagation? Maybe last question for me, for Ibu Yalni. Hello. Okay, Ibu Yalni, can you hear me? Okay, maybe maybe she has a little problem with the connection, I think. Okay, maybe she has a problem. So is there any other question? Because actually the time is for the question and answer is already finished, I think. If there is another question, I think we can wrap it up. Thank you very much, everybody, for very nice presentation. So I hope you have a very good day today, and I hope you have a keep in high spirit because we still have another couple hours ahead. Okay, and once again, thanks everyone. Have a nice day. I'll go to
Thank you.
the main conversation. Thank you very much.
Thank you very much.
Thank you all. Thank you very much.
Thanks all.
Recording in progress.
Would you like me to already start sharing my screen? Would you like me to start sharing my screen already?
Not yet, sir. Or you want to try the technical matters right now?
Yeah, but I think, I can just try for a second.
Yeah, you can try before we start again at 3.
Just a second.
Yes.
Yeah.
Let's see.
Yes, we can see your slide clearly.
Yeah, that's it. That's fine.
Yes.
Okay, I'll stop sharing now then.
It's
and
okay.
you can put it on
you.
again. And let me see. Stop sharing. Yes.
Okay.
Good.
Thank you, Dr. Arnold. Menit lagi. Ladies and gentlemen, so happy to welcome you back on the main room. So the next agenda is plenary sessions. Therefore, may I invite Dr. Hayati Iskandar as the moderator of the sessions. But before that, I would like to read short profile of her. Dr. Hayati Minarsi Iskandar is a researcher of Indonesian Oil Palm Research Institute who are interested in molecular biology, biotechnology, and biochemistry. After graduated from Bogor Agricultural University or IPB, she continued her master degree in plant molecular biology at Vrije Universiteit Brussels, Belgium, and obtained her PhD degree in biochemistry and molecular biology at the University of Queensland, Australia. Without further ado, please welcome Dr. Hayati Iskandar. Dr. Hayati Iskandar, time is yours to lead the sessions.
Okay. Thank you, Ibu Aulia, for my introduction. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon in Indonesian time, distinguished guests, all the speakers, and all participants. Welcome to the next plenary session of the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBio 9 2022. Please allow me, Hayati M. Iskandar from IOPRI Bogor, as a moderator to lead this session. This afternoon, initially, we will have two guest speakers, Dr. Arnaud Bovy from the Netherlands and Professor Chin Kun Wang from Taiwan. Unfortunately, Professor Wang is not able to attend this session because he suddenly has to conduct a surgery in a hospital. So we will start the plenary session with the presentation of Dr. Arnaud Bovy. Am I right to pronounce your name, Dr. Arnaud?
Yes, you're right.
Okay,
Absolutely.
thank you. First, I would like to introduce you about Dr. Arnaud Bovy. So his CV. Dr. Arnaud Bovy, he's a researcher at Department of Plant Breeding, Wageningen University and Research. He's also a group leader at Breeding for Quality Research Group, also at Wageningen University. And his research interest is metabolomics, plant breeding, and genetics. So Dr. Arnaud will present an interesting topic about application of genetics and metabolomics in assisting plant breeding for quality. So you will have 20 minutes for presentation, and we will have a Q&A session after that. So Dr. Arnaud, the time is yours. Thank you.
Thank you very much. I will now share my screen. Okay, well, good afternoon, everybody. It's a real honor for me to be invited to give a talk at this conference. I really would like to thank Wayuni and the other organizers for inviting me here. And well, the title of my talk in today's session is Application of Genetics and Metabolomics in Assisting Plant Breeding for Quality. And so, as was already mentioned in the announcement, I lead a research group on breeding for quality within the plant breeding department of Wageningen University and Research. And in my talk today, I will talk about the combination of genetics and metabolomics in order to assist breeding for quality. So we are in the plant breeding department. And well, actually, I would like to start with the question, what breeders want? What do they want in their breeding? First of all, they would like always to find novel sources of their traits. And in particular, they are looking for genetic variation, and in particular, crossable genetic variation with which they can, let's say, improve their crop with traits of interest. Breeders also would like to have tools to select for their traits. And on the one hand, these are efficient phenotyping tools, but more and more important in breeding is to have genotyping tools so that breeders can already select for the trait of interest at seedling stage by using molecular markers. And important for this story is that the best possible molecular marker actually is the gene that's underlying the trait of interest. In addition to breeding for yield and disease resistance, there's a growing interest to also breed for quality traits, and that's the topic of my work. And if you look at these quality traits, you can basically look at or see a number of traits that you can visually monitor, such as size, shape, color, and shelf life of your crop. But there are also invisible traits of interest, like nutritional value, taste and aroma, and pungency. And especially in order to monitor these invisible traits, we need a good detection platform. And many of these quality traits are determined by small molecules that are present in our fruits and vegetables, small metabolites, and detecting these metabolites is one of the main aims of our research. Well, we do that using metabolomics, and metabolomics is a technology aimed to provide a broad overview of the metabolic composition of a given extract. And metabolomics, as we use it, is based on mass spectrometry, which basically stems from the idea that every molecule has its own specific mass and can be detected as such. Well, what are the advantages of metabolomics for breeding? First of all, metabolomics can give you the insight in the metabolic basis of often complex traits. It also allows you to define subtraits. And defining subtraits is a way to simplify genetics, as I will show you in a minute. And also metabolomics and knowledge of pathways helps you in identifying genes that may be involved in this process. And to illustrate that, I would like to give you an example of a complex trait, taste, a trait that we worked on a lot in particular in tomato. And when you want to study the genetics of taste, the first level you can do that is by measuring taste appreciation using consumer panels. But you can imagine that just giving an evaluation of liking is not giving you a lot of differentiation in the traits. So a second level at which you can look at taste is at the level of trained tasting panels. And these trained tasting panels, they are able to discriminate many more aspects of taste. For example, they can dissect taste into, for example, taste, aftertaste, mouthfeel, and aroma attributes. And under these attributes, there are different sub-traits like sweetness, sourness, umami, and so forth. This is already a new level of detail. But if we would look at the metabolites underlying these traits, we can see that under each of these attributes, there are multiple players that contribute, multiple metabolites that contribute to this particular trait. So in fact, this very complex trait, taste, can be divided in many different subtraits by looking at the metabolites. And looking at these metabolites, studying these metabolites, also gives you hints on the biochemical pathways that are important and the genes that may be involved in different steps of these pathways. So that's a big advantage of studying metabolomics. Well, in addition to metabolomics, today's breeder's toolbox has really expanded. And it has expanded, for example, by the availability of genome sequences. And if you look at a crop like pepper, more than 400 accessions have already been sequenced today. In tomato, it's even more than 700 at the moment. There are high throughput marker systems available to genotype large numbers of genotypes. Transcriptomic tools and databases are available, and of course the metabolomics platforms. In addition to these tools, also there are more and more interesting breeding materials available. For example, the presence of mapping populations that you can use to study the genetics of your trait. A specific type of mapping population that I'm going to talk about today is a GWAS panel, which basically is a diversity panel of accessions. Tilling populations are available nowadays, so mutagenized populations that you can use to identify mutations in specific genes of interest. And of course, transformation technology, which is available, very important to, for example, study genes by virus-induced gene silencing, or make stable knockouts using CRISPR-Cas gene editing. Well, in my talk today, the high-throughput market platforms, metabolomics, and the use of GWAS panels is going to play an important role in the example I would like to give. It may be good to mention, in my research, tomato and pepper are the major crops that we work on, and the talk today, as you can see from the pictures, will be about pepper. The topic of my talk today was studied in a big EU project called G2P-SOL, Genotype to Phenotype SOL. It is a large EU project that just ended at the end of last year. And the aim of this big program was to link genetic resources, genomes and phenotypes of solanaceous crops, tomato, pepper, potato, and eggplant. And my involvement was mainly in the pepper part of this work. The aims of this project was to do an inventory and conservation of material stored in gene banks. And it was mainly done by high-throughput genotyping. I will come back to that in a minute. A description, assessment, and phenotyping of the collection, genetic analysis of this phenotypic variation, and there was a pre-breeding part in this program. Well, I already mentioned that our main involvement was in pepper, and as I already mentioned, also genetic diversity that was studied is the diversity stored in gene banks. And for each of the crops, more than 10,000 different accessions were genotyped using genotyping by sequencing or other next NGS-based platforms. So for capsicum, 10,000 different capsicum accessions were sequenced. Most of them were capsicum annuum accessions, but also a number of accessions from Bacatum, Chinense, and Frutescens. And let's say the genotyping and the insights we gained about the evolutionary history of pepper has been published in this nice paper of Pascale Tripodi in PNAS last year. Well, based on the genotypic analysis and the genotypic diversity that was found, a core collection was made consisting of 450 capsicum annuum accessions. And this core collection was studied further as part of the project. And because this core collection was derived from the genotypic data, it was expected to represent the genetic diversity within capsicum annuum. And this core collection was further analyzed by high-density genotyping and by phenotyping of 3 trials conducted in different countries in Europe. Well, here you just get a small overview of the phenotypic variation that was present in this collection. So there's a huge amount of variation. Now, what we tried to do in the past year was to get a better understanding of the metabolic diversity in this pepper core collection. And if we think about the metabolic composition of pepper fruit, actually pepper makes a lot of very interesting compounds. Compounds involved in color formation, like carotenoids, chlorophyll, and anthocyanins. Compounds involved in providing pepper with taste and aroma, like volatiles, sugars, acids, and capsaicinoids. And other nutrients, dietary nutrients, like vitamins, capsaicinoids, and capsiates. So we were aiming to measure these compounds in this pepper core collection. Now, the trial that we conducted looked as follows. The collection consists about 400 capsicum annuum accessions, 20 Chinenses, Frutescens, and Bacatum collections. They were grown at different locations, and I will talk today about material grown in France and grown in Italy. And ripe fruits of these trials were harvested and subjected to different metabolomics analysis. So basically, LCMS to detect semipolar secondary metabolites, and GCMS to detect volatile metabolites. Now, how many compounds can we detect using these platforms? Now, if we look at the GCMS platform that detects volatiles, we detected 128 different volatiles in the INRA trial in France and 133 metabolites in the trial in Italy. And most of the compounds that we detect, we can actually identify. And for the GCMS, we only had, let's say, 10 unknowns in total. With LCMS, it's a completely different story. In LCMS, we were able to detect, let's say, hundreds of compounds, but the majority of these compounds actually are unknown, and identification of compounds using LCMS is still a big challenge in metabolomics research. And the groups that we could detect consisted of capsaicinoids, flavonoids and other phenolics, and capsyanosides, which are diterpenoid compounds. Now, if we look at the distribution of these metabolites over the different accessions, you can visualize that using a principal components analysis. And in this principal components analysis you see here, actually all the dots represent the different accessions in our trial, and with the arrows, the main drivers of the variation are shown. And you can see a clear division in the first axis of the pungent genotypes on the right side and the non-pungent capsicum onion peppers on the left side. And if we look at different, let's say, volatile compounds and taste-related compounds, we can see that on the right corner here is the place where most of the wild accessions end up, and they are, let's say, the richest in volatile esters, which usually give a fruity aroma to your fruit. So this gives you some insight in the metabolic variation, in this case for volatiles in this material. Now, there are hundreds of compounds that we can detect, but which are the ones to focus on? Which are the ones that are important, for example, for taste? Well, one way to do that is to see if you can find correlations between the presence of certain compounds and a certain taste attribute. And using a few colleagues, we basically determined the aroma and the taste of all these peppers. And we could discriminate, for example, 3 different types of aroma. The beany green pepper aroma that you find mostly in blocky peppers, a more grassy green fleshy cut grass aroma, and a sweet fruity aroma. And when we look at the correlations of these taste attributes with different metabolites, we can clearly see that methoxypyrazine is the compound that really is responsible for this beany green typical blocky pepper aroma. If we look at the grassy green type of aroma, it's mostly the lipid-derived volatiles that are highly correlated with this type aroma, and that is very well in accordance with also taste in tomato, where these compounds also play a very important role. And if you look at the sweet fruity aroma, it's especially a number of volatile esters that are highly correlated with sweet fruity aroma. So in this way, we can basically select a number of potential target compounds that we could use in breeding for taste. And for example, if you would like to breed for a pepper with a more sweet and fruity aroma, we would aim at increasing the amount of esters and decrease the amount of these methoxypyrazine or oxylipin-derived volatiles. So these could be potential breeding targets for taste in pepper. Well, we also tried to see if we could link the metabolic variation that we see to genetic variation. And to do that, we used a GWAS study, and GWAS stands for Genome-Wide Association Study. So basically what we try to do using molecular markers is to find associations between specific molecular markers and specific compounds. So the data we used for this genome-wide association study were these volatile aroma compounds and the secondary non-volatile metabolites, around 200 metabolites in total. We used the data from the 2 trials, from the 2 different locations, and we used SNP data from genotyping by sequencing, and we used in total 10,000 SNPs in this analysis. Now, we used also 2 different, let's say, standard approaches that are used in GWAS, mixed linear models and a blink approach. Now, in the GWAS, we could find a number of common QTLs that were found in both location trials. In total, we found QTLs for 105 metabolites at 524 different loci. And the first one I'd like to show you here is the GWAS analysis. GWAS results often are visualized in this kind of what we call Manhattan plots that here show you the distribution of the 12 chromosomes of pepper, and here the association of your compound with specific loci on the different chromosomes. And you can see here a very nice association at the end of chromosome 2 of capsaicin, the compound responsible for pungent taste in pepper, and this location on chromosome 2. And it's known that this location is the famous PUN1 gene, which is the gene that's the ester-forming enzyme responsible for the last step in the biosynthesis of capsaicin. And this very nicely matches with earlier results in which gene bank annotations for pungency also showed up a very nice QTL at exactly the same spot. Another observation that we did, and relevant for this story, is the fact that biosynthetically similar compounds, they correlate quantitatively with each other. And to show you that, we made this, let's say, this hierarchical cluster here, and each of the colors actually represents metabolites that are closely related to each other. And you can see that these metabolites form rather separate clusters. So it means that metabolites with the same origin or the same biosynthetic pathway are often correlated with each other, which would suggest that the regulation of these pathways may be regulated by hotspot QTLs. And the question is, do we see this kind of hotspot QTLs? And actually, when you look at this graph, here, this is actually a merger of all the QTL data that we found in our study. And we try to visualize these data using bubble sizes. And the larger a bubble size, the more compounds are actually co-localized on the same QTL. So, for example, if you look at the bubble size here, at the position of the QTL for capsaicin, actually, we have here not only one different capsaicin, but a number of capsaicins that show the QTL at this particular position. And that is because this ester-forming enzyme, PUN1, does not only use one specific substrate, but has a broader specificity for different substrates, leading to different capsaicinoids. The same type of very nice bubble here, we see a QTL for different flavonoids. And we see this QTL at the end of chromosome 5. And our most associated marker was landing exactly in the MIP12-like transcription factor. And MIP12-like transcription factors are actually known to regulate the flavonoid pathway. And we cloned the MIP12-like gene from tomato a number of years ago and showed that these transcription factors indeed are regulating the flavonoid pathway. Well, actually in pepper, we find a very nice candidate gene called MIP12-like, which seems to regulate also the flavonoid pathway in pepper. The MIP12 gene in tomato is actually located on chromosome 1, and this ortholog is located on chromosome 5 of pepper. So we found quite a number of nice and interesting QTLs. But one of the main challenges of GWAS studies is to validate these QTLs, to make sure that they are really showing you the right lead and not false positive, for example. And one of the QTLs we validated was actually this MIP12-like QTL I was just talking about. And it was very nice to validate this QTL because we started that work already quite some years ago. And it was actually the work of Wayuni, and here you see a picture of Wayuni 10 years ago when she was still working in Wageningen, and she identified 2 accessions We call them here for simplicity P12 and P24. P12 was a very high flavonoid accumulating accession, and P24 was a very high vitamin E accumulating accession, but low in flavonoids. And in collaboration with Ilan Paran in the Volcani Institute of Israel, we developed segregating populations based on the cross of these two parents. And when you make segregating populations in the F2, these nicely segregate, and every F2 individual basically is a genetically unique individual. So we did a QTL analysis of this F2 population, and actually in this population, we also find a very strong QTL at the same position on chromosome 5. This QTL, you can see here that if you have the P12 allele of this QTL, you get much higher flavonoid levels than when you have the P24 QTL allele. And also what we found was that a large number of flavonoid genes showed a much higher expression when the P12 allele is present compared to when the P24 allele is present. And this, of course, suggests that there is a transcription factor into play here. And actually that also led us, independently of the GWAS trial, to the identification of the MIP12-like gene. Now, we did a number of experiments to indeed validate and show that this MIP12 gene is indeed the gene doing the job. One of the things we did is we knocked out the MIP12-like gene in this parent 12, the flavonoid accumulating parent, using virus-induced gene silencing. And you can see if you have the flavonoid level in the control, here plants that in which we used a Gus construct to silence, versus the MIP12-like silencing, you can see a clear decrease in flavonoid content in the silenced plants. We also try to overexpress the gene in a pink tomato background, so in a MIP12 mutant of tomato. And also here you see that when you have high expressors of the MIP12-like gene from pepper, you can see a clear increase in flavonoid content compared to the low expressors or the controls. And last but not least, very important for breeders, is can we use this gene actually to increase the flavonoid content of commercial materials. And we did that in a few backgrounds, and I'll show you here the results of introgression in sweet banana. And you can see when you introgress the P12 allele, you really get a nice increase in the flavonoid content in sweet banana compared to, let's say, the control without this allele. So also for breeding purposes, this gene is a very nice marker, and P12 is a very nice donor for this trait. Now, the ongoing research is actually focused on validating QTLs that we found in the GWAS in biparental segregating populations. We are mining these QTL regions for candidate genes and hope to identify and analyze candidate genes. We also want to improve our GWAS study because our study was really limited in the number of markers. I already mentioned we use 10,000 markers, but this is far too low for a GWAS study. And that means that we are certainly still missing maybe important QTLs because we don't detect the right alleles. We are trying to alleviate this problem by sequencing the entire core collection, and this is something that will be done in the near future. And finally, we'd like to also combine different data types. For example, we would like to combine our metabolites with taste, as I showed you, but also with, for example, shelf life data. Now, finally, I would like to come to the conclusions. I think that gene banks harbor a very wonderful resource of genetic variation that can be exploited by both researchers and breeders. The breeder's toolbox has really expanded in recent years with various exomics platforms, making it possible to have a much more detailed look and getting detailed insight in our traits. I hope I showed you that metabolomics is very useful to get insight in the metabolic variation in a crop, in the metabolic basis of traits, and in the genetic basis of traits. And finally, GWAS provides novel donors, QTLs and markers for traits of interest. But an important part, and that will take years to come, is the validation of the QTLs that we found. So last but not least, I would like to acknowledge the people involved in this research. From my own team in Wageningen, Yuri Tikhonov, he plays an important role in all the research we do. Jos Molthoven and Fien Meijer, two technicians in the project. Yi Wu, a PhD student who participated in this work. And then Richard Vinkers, Matthijs Brouwer, and Giorgio Tumino, who were involved in the statistical analysis of the GWAS data. We collaborated with people in France and in CREA to set up these GWAS trials, and with Ilan Paran from the Volcani Center, in which we collaborate closely in this research. And finally, all the partners that participated in G2P-Sol, who made it possible to have such a large-scale project going on successfully. And this is where I'd like to end my presentation, and I'm happy to answer some questions. Thank you.
Thank you, Dr. Arno, for the nice presentation. So, ladies and gentlemen, now we will have a question and answer session for about maybe 10 minutes. So you can deliver your question by chat box or raise hand. So please, you can start to give questions to Dr. Arno. Okay, now I have one from the chat box.
Yeah, very nice question indeed.
Okay.
For a human tasting panel, won't capsaicin ruin your taste bud to differentiate other tastes and aromas? Yeah, it's a very good question, and especially for Western people, it's of course very difficult to taste anything once you taste capsaicins. So if you talk about aromas, it's not so problematic because you don't taste it, but you just smell. But for really the combination of tasting and smelling, we cannot do that ourselves. And we did that in the past using actually Indonesian people to do the tasting, because they can much better differentiate tastes in the background of high pungency.
Okay, thank you, Pak Dani Satyawan from BRIN. Is there answer your question?
Yes, and it's quite amusing actually to find out some Indonesians actually have some use in this kind of studies. Thank you, it's very amusing.
Okay, thank you. Next, please, for the next questions. Or maybe I can also ask you, Dr. Arno.
Yes.
I'm interested when you said that metabolomics or metabolic is basis usually used for the complex traits. So what about the, like disease tolerant or resistant? Yeah, because we know in Indonesia that are the big problems, especially for this chili pepper. You know, Indonesia very love chili pepper, chili fruit or chili pepper very well. So maybe you can explain about this. Can we use also this metabolics?
No, certainly this can be done, and we have very nice examples of it also. So also in the basis of resistance, metabolites play a role. And indeed, metabolomics can be used to get a better insight in the mechanisms that the plant uses for resistance against pathogens. And especially also when you want to, for example, get insight in the type of genes involved, a very detailed insight in the structures and the metabolites that are playing a role there, it can really help in identifying the underlying genes.
Okay.
Yes, it's definitely a very nice combination to combine resistance and metabolomics.
Okay, thank you, Dr. Arno.
Yes.
I see that Dr. Wahyuni is raising hand. Please, Dr. Wahyuni.
Yeah, thank you, Bu Hayati.
Yeah.
Hi, Arnold. Thank you for including my pictures in your slide.
Well, you made the basis for our paperwork, as you know. So.
Yeah, I amazed with your G2P Sol project. It's a very big project, including at least 400 capsicum annuum accessions, in addition to Pakatum, Chinensa, and other species.
Yes.
And I saw the PCA of the volatile compounds. It's ended similar. I mean, the volatile compounds with this big hundreds of accession, they differ the accession based on the pungency level. Like we have in the last time that we used 32 accessions. Yeah. And then with this project, have you already identified whether these volatiles came from or derived from the capsaicinoid pathway?
Yeah.
And yeah, the second question, Arnold, or later.
Shall I first answer your question? Your first question?
Yes, please, Arnold.
Because you know, this is a very interesting question, because what we indeed see is that pungent accessions make a lot of volatiles, specific volatiles, as you only find in the pungent accessions. And we don't know if this is, let's say, due to really a genetic, for example, the PUN1 locus, whether that is also involved in producing specific volatiles, because it's an ester. PUN1 is an ester-forming enzyme, so basically you could expect that it can also, for example, produce volatiles, or whether this is due to the fact that it's just an association, because these are two different sets of germplasm, the hot germplasm versus the non-pungent germplasm. Actually, we still don't have the real answer to that question, Juni. We had the question already in the time that you were working in Wageningen, but it's still an open question. And but we know what type of experiments to do to show it.
Okay.
So we really need a PUN1 to make a PUN1 mutant, an isogenic PUN1 mutant that in a pungent background, and that I think will enable us to answer that question.
Okay, so this one of the complex traits, right? These volatiles.
Yeah, sure.
Yeah.
Yes.
And
no, so we are very interested in answering that question, but didn't do the right experiments yet to answer it.
Yeah. That's one question that I want to ask you. If we have this complex traits, and can you suggest, or well, at least if you have something in mind, what kind of experiment that we should design to answer some kind of, for example, the markers underlying these complex traits?
Yeah, I think the problem with complex traits is that genetic studies are difficult because the complexity is too large, and you don't have the statistical power to detect all the players that contribute to your complex trait. So that's why we think it's important to really define subtraits, which we do based on, for example, metabolites, and then study each of these subtraits, because it will make genetics simpler. So that's one thing I would advise to do, because if you only look at the phenotype of the complex traits, probably you're not going to get there. So getting insight in your trait is very important, and so metabolomics is one way to do that.
Yeah,
is that answering your question?
Yeah, well, yeah, metabolomics.
And then of course, the next step is when you are able to identify, for example, markers for different of these subtraits, of course, you have to prioritize them based on the contribution that they make to the final outcome of your complex trait. And that is something you have to evaluate, for example, from your QTL analysis to see what is the percentage explained variation for each of these different subtraits. And then you can focus on the ones that are giving you the best possible outcome.
Yeah. Okay, Arnold, thank you.
Okay, thank you, Bu Wahyuni. So we still have now from the chat box from Bapak Dylan Lowry.
Let's see.
How would you plan to approach the unknown compound that detected in your LC and GCMS analysis?
Yeah. So as I already mentioned, in GCMS, it's not such a big problem. We have only a few unknown compounds, but in LCMS, that's really a problem. Now, we don't aim to identify all these compounds. That sometimes it can take a PhD study to identify a few compounds. It can be very difficult. That's why what we do is we use these unknowns in our studies, but only when we can see that an unknown compound really contributes to a trait of interest, it is worth for us the effort to try to identify it. And the way to identify it, for example, is to do a much more elaborate LCMS study with MS fragmentation in order to really find out what the structure of your compound is, just as an example. But our approach is very on purpose. Don't bother too much about the identification in the beginning, but do that at the end when you know that these compounds are really important.
Okay. Mr. Dylan.
That was, I think, the answer to the question. Yeah.
Okay.
Then how would you know that the unknown is gonna contribute significantly to the traits?
Okay, so well, one way to know that is to do a correlation analysis to see if there's a correlation between the presence of your compound and your trait of interest, as I showed you for example, for taste. And when you have a certain taste attribute and you look at the correlations of all your metabolites with this particular attribute, that's one way to, for example, look at this correlation. But there are many different ways to do that. One other way that for example, we like very much at the moment is when you are interested in taste and you run a GCMS analysis, one of the things you could do is to connect your GCMS device to an olfactory port and smell each of the compounds that comes off your GC column. And in that way, you can also detect specific flavors and detect specific compounds. And what we have seen is that sometimes compounds that are highly detectable and give you very small signals can give a very powerful taste experience, and also the other way around. So this is another way to find out, okay, I don't know the compound, but this one we need to focus on.
Okay, Dr. Arno, we still have one more question, and maybe this is the last question for this session from Dr. Penny Lestari, or Ibu Penny Lestari. So, on the level of pungency of chili is also influenced by the environment, such as dry and other stress. Okay.
Yeah, this is an interesting question. It is known indeed that capsaicins are strongly influenced by the environment, and I know that breeders are very interested in ensuring that they have a stable amount of capsaicins in their crop that's less influenced by the environment. But we don't, I at least don't have the knowledge whether how big that influence is and whether this indeed changes with shelf life. It's a good question, but I don't have the answer.
Okay, so Bu Penny, maybe we can further discuss with Dr. Arno after this session. Okay, So thank you for the fruitful discussion. Oh, there is one more. Maybe Dr. Arno can answer with a brief question. These questions in briefly.
So which is the most reliable result between genetics and metabolomics? I don't know exactly what you mean with this question, because I think it depends. Reliable for what? So that's hard to answer. But both platforms have their pros and cons, and I think that especially the combination is strong. If we, for example, see that compounds that are correlating also end up at the same regions, also genetically show the same QTLs and so on, well, this gives us confidence, for example, that a certain compound may be involved in a certain trait.
Okay, thank you, Ibu Rita.
So it's basically using the correlation on the one hand, genetics on the other hand. If everything points in the same direction, that gives you a certain level of reliability.
Yeah, but metabolomics also needs the genetics information, right?
Yeah, you know, when you want to do metabolomics and you would like to look at variation, or you need variation in order to see differences, and our approach is always based on detecting differences. And there, you know, having, for example, genetically diverse material is a must. Another way to create diversity, of course, is different treatments. We have focused much on using genetics, but you can also use treatments. But especially also in relation to the question of the identification of compounds, when you don't have variation, it's also impossible to, for example, prioritize compounds to focus on. So that, I think, discriminates also metabolomics from, let's say, the more classical biochemical studies that are really aimed at identifying every compound. Now we are really focusing on the ones that do the job, that are important for our traits.
Okay. Thank you for the questions. So, and also this fruitful discussion for Dr. Arno. So maybe in the future, if we can see from your presentation, this metabolomics and GWAS or genome-wide association study will be more improved, and many people maybe use it for in breeding session, right? Okay. So again, we appreciate Dr. Arno Bovi for the excellent presentation. Maybe by giving him a big applause. Thank you for your attention also, everyone, and I will close this session. For the next program, I'll return to the committee or the Master of Ceremony. Thank you again, Dr. Arno.
Thank you too. It was a pleasure.
Thank you.
Thank you, Dr. Arnold, for the interesting materials. And also thank you to Dr. Hayati for leading the sessions this afternoon. Very fresh, ladies and gentlemen. So for the next agenda, we are going to announce best oral presentations. Who is the best oral presentations during 2 days of ISIBio? Any guess, ladies and gentlemen? And the best oral presentations goes to, drum rolls, any guess? Yes, congratulations to Dr. Sugiono Saputra from Research Center for Applied Zoology with the title of Sago's chart hydrosate as a carbon source for expolysaccharide productions by Lactobacillus plantarum. Congratulations for Dr. Sugiono as the best presenter at the NIN International Symposium of Innovative Bioproductions for Biotechnology and Bioengineering, ISIBio. Please give a round of applause, everyone. Ladies and gentlemen, and now I would like to invite Mr. Ahmad Fatoni, PhD, Head of Research Center for Applied Microbiology, National Research and Innovation Agency, to convey closing remarks. Bapak, screen is yours. Still unmute.
Yeah, sorry. Yeah. Yeah, thank you, Ms. Aulia. So first of all, I would like to thanks here still we have our speaker, Dr. Arnold Bovi. It was very interesting talk, and then many questions. I hope that it can be follow up by the collaboration with him in the near future. Okay, everyone. Assalamualaikum warahmatullahi wabarakatuh. Good evening, grace and peace, everyone. I'm Ahmad Fatoni, representative of Dr. Iman Hidayat, Chair of the Research Organization for Life Sciences and Environment BRIN. It has been a pleasure to be with you since yesterday in the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022 with the theme of strengthening bioeconomy through applied biotechnology, bioengineering, and biodiversity. Thank you everyone again for attending this symposium. On behalf of the Research Organization for Life Sciences and Environment BRIN, I would like to express gratitude to all keynote speakers, invited speakers, presenters who have shared their idea, knowledge, and finding with us during the symposium. Since yesterday, I observed the excellent presentation from the great work of everyone and active discussion among our speakers and participants. So this symposium covering 6 topics, including bioresources and biodiversity, nutraceutical and natural products, biotechnology and bioinformatics, medical sciences, agricultural and forestry, and the last, environmental sustainability. So therefore, I can conclude that the purpose of our symposium this year has been accomplished. But however, I hope that this symposium is not just a tool for us to share our finding or idea and stop with the publication, but bear in mind that we still have a big responsibility to implement our findings to our any potential collaborators or stakeholders such as society, industries, and other users. Hence, our research hopefully can make a significant impact to the economy, society, and the nation or Indonesia. And one of the key success to achieve enough impact is collaboration. And I would like to take this word of collaboration as a key home message to all of us. Implementing our results and making an impact are not easy tasks, but I believe we are going to make it by working together with others. As our chairman of Research Organization for Life Sciences and Environment BRIN, Dr. Iman Hidayat mentioned yesterday in the opening session, that there are 3 factors that we should have done. Actually, first, working together with colleagues from other disciplines. The second, working closely with industry, and the last, mastering key technologies in order for us enable to utilize our biodiversity sustainably. So I do really hope that after this symposium, everyone, we would extend our collaboration with other colleagues that we just met during this 2-day symposium, or even with our keynote speakers from Indonesia or overseas. And here we still have Dr. Bovi. I hope that in the near future, or Dr. Wahyuni already have collaboration with Wageningen and others. Hopefully we can strengthen the collaboration with the Wageningen University. And I believe also that researchers and participants here already have their own collaboration, but I think there should always be a room to enhance our collaboration. Okay, before I close my talk, congratulations to Pak Sugiono, who have won the best presenter, and hope it will give motivation to us to get better and better in the future. Lastly, I would like to thank the entire committee of ISIBio 20. Oh, sorry, stop my video. Yeah. Can you see my video? Yeah, lastly, I would like to thank the entire committee of ISIBio 2022 that have done a fantastic job. They have spent their time, thoughts, and energy to prepare all of this. Thank you, Dr. Nani, Dr. Wahyuni, and all team members for making this symposium a successful one. Also, thank you to our collaborators, Indonesian Biotechnology Consortium, Consortium Biotechnology Indonesia, of Indonesia, and other who have supported this event. Again, to all participants, thank you very much for your attending in ISIBio 2022. And finally, I would like to close my remarks and officially end the 9th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering 2022. Wishing you all a success and hope to see you in the next ISIBio next year, inshallah. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Stay safe, everyone. Thank you.
Thank you
I
for the
give back to
remarks and also officially closing ISIBio 2022, Dr. Ahmad Fatoni. Ladies and gentlemen, that was the last agenda of these 2 days, ISIBio 2022, as well as mark the end of ISIBio itself. On behalf of official committee, I would like to sincere highest appreciations and also thank you so much for your attendance and active participations in all sessions during the conference held. See you on the next ISIBio next year. Stay safe and healthy.
Okay, thank you everyone. Yeah,
Thank you.
do all the best. And see you next year. Yep. Thank you.
Can you still hear me? Hi Anna, thank you for
Hey,
giving
Uni.
lecture today.
It was a pleasure. I hope it was what according to your expectations.
Yes, yes, I know, of course.
No,
Thank you
but
so much,
no, it was
for the time.
it was nice to do and nice to see you again.
Yeah.
Recording stopped.
I guess it was a very nice conference. Eh? I have not heard much about it. Of course, it was during the night for us, but it's very good that you are organizing this. How are you doing apart from this uni?
Still doing the research in paper about the post-harvest.
Yeah, I did not talk about it, but I told you, I think, we also are working on post-harvest at the moment in pepper.
Yeah.
So this same collection that I talked about, we also did a shelf life trial with it to see how much variation we see in shelf life, and there is considerable variation that we are actually exploring further at the moment.
Okay. But have you seen session that we have or has this delay in fruit softening, for example, or retain their freshness after?
Yeah. Surely see, yes. So what we did is we mainly looked at water loss. So we just did it by weighing, you see quite a big diversity in this trait. We also did the GWAS on these results. We have a few identified a few loci that could be interesting, and we are now actually making segregating populations and looking more closely at the number of selected accessions to see if we can let's say get a step further, get more insight in what's important here.
Yeah, I know. Continue with this analyzing the markers